virtual presentation by Sharon Hutchinson, and she's going to talk about shrimp trawl discards.
Is on future earnings lost due to shrimp trawl discards, saving from a bycatch reduction trial. I'm Sharon Hutchinson. I'm a natural resource economist at the Department of Agricultural Economics and Extension at the University of the West Indies in St. Augustine, Trinidad. Thank you for being here with me. And I'd just like to go over background to the problem, the objectives and methodologies used, the key results, and what can we take away from the work that was done. Firstly, I'd like to say that this work was conducted in Trinidad and Tobago, which is the southernmost Caribbean island, just next to Venezuela. And in this space, shrimp trawling is done primarily on the west coast in the Gulf of Paria, on the southern coast in the Columbus Channel, and a bit in the north coast. And it's done year-round. Now, the shrimp trawling that is happening here is using traditional trawling gear, and this gear drags along the seabed and damages the seabed, and so it's considered to be unsustainable by all the stakeholders involved, including the fishers. The trawls, in addition, catch a large proportion of bycatch, and FAO estimates this to be 3 to 15 times the target catch. And from what we know of this fishery so far, the bycatch share is normally between 80 to 85% of the total catch. It is a small-scale fishery with approximately 340 fishers, stemming from what we consider to be artisanal, very small-scale, non-mechanized vessels, which is about 160 vessels, and the larger vessels, which are what we consider to be non-artisanal, which are using mechanized gear, accounts for most of the other fishers. And here we're just talking about 140 vessels in total. Now, shrimp is a big part of the total catch, estimated to be about 5%. And I say big part because it is still a single species. Taking up 5% is important because there's so many other species of seafood caught. What is noteworthy here is that since the late 1990s, there's been a generally upward trend in the share of shrimp as a proportion of all seafood. And the fish, the shrimp and the bycatch are normally caught in Orange Valley and Tahiti, which are on the west coast of Trinidad, and sold at wholesale markets that are oftentimes near to the land sites, and also by fish vendors at the land site, and also in municipal markets and via mobile vendors as well. So the key objectives of this study was to determine the composition of bycatch coming out of this bycatch reduction trial. So we wanted to also determine the composition of the discards, because some of the bycatch are marketable fish species, some are thrown out, and then we wanted to calculate the amount of earnings that are lost in the fishery due to the discards, looking at the difference between what was caught from the net that had the BRD device versus the control net, and then analyze the differences in terms of how much marketable shrimp was caught, the marketable bycatch, and the discards as well. So there were 3 trials done in one in 2017, one in 2019, and the other one in 2020. And in these trials, the nets on both sides of the vessel and the BRD device was attached to one, while the traditional net was used in the other for each hole. And coming out of each hole, a sample of the catch was taken, and from there, there was a weigh on board of the shrimp, the marketable bycatch, and the discards. And so that data was used to extrapolate to the total weight of each hole. The analysis, the data was collected for the holes, which was more than 20 holes in each season, taking place on a, what we call a type 4 commercial vessel. This is the largest vessel type for this fishery. And the data is analyzed in Excel. And based on wholesale prices obtained from May 2022 in Trinidad and Tobago, we're able to calculate the value of the loss share. And based on the data from the holds, also calculate the share of the marketable bycatch shrimp and discards. And then we also compared the discards from the traditional net as well as the net with the BRD device to see whether there were changes in the average share of shrimp, marketable bycatch, and so on for each of these polls and each of these trials. So what was found is that most of the discards were occurring for two main species. Firstly, pan salmon, which accounted for 29% of all the discards, and this would include the juveniles and the adults that were thrown out. And then this was followed by Bangamary, which accounted for 25% of the discards. And here on the left, you'll see essentially a bucket of your bycatch species is very mixed. There are catfish in there and other species as well. And so the shrimp will be separated from the bycatch, and fishers will bring it on shore in these plastic buckets and then take this to the wholesale market. In terms of the marketable bycatch, however, you'll find that the main marketable bycatch is what is locally known as crow, and that is accounting for 35% of all the marketable bycatch. And Bangamary is also accounting for 28% of the marketable bycatch. Now, the Bangamary occurring as a marketable bycatch in this large proportion is really significant because while a lot of it is being brought to market, it's accounting for 28% of what's brought in as the marketable bycatch. In fact, if you were to go back and look at the discards, it's also accounting for a significant amount of the discards. And here the discards are in fact mostly juvenile fish that are being discarded. This would be another depiction of how the fish species are sorted when it comes to the wholesale market. So the shrimp, and then they're sorted by species, because of course each species will get a different price. And these type 4 trawlers are normally between 40 and 50 feet in length. And the fishery is estimated to have 25 of these vessels that take average trip of 7 days each, 3 holes per day. So based on the fishery profile, what was concluded from the trials is that the BRD device significantly lowered discards. And this is at a 5% level of significance relative to the traditional control net. In fact, in 2019, the BRD net reduced discards by 47%. And based on this, it was estimated that compared to the traditional net, a fisher would save 48% of their earnings if they use this BRD device. And how was this calculated? Well, in general, just in summary, what was done was that we looked at the adults that were discarded. So this would be the rightmost column here. And so for a year, the fleet would have lost $0.7 million in earnings by discarding adults. And then of course, for the BRD, and then the BRD in terms of what the juveniles that were discarded, those juveniles accounted for $59.6 million. So in total, you would see that what was lost by using the BRD net was approximately $60 million. Now, compared to the control or traditional net, in fact, which accounts for almost twice as much would have been lost to juveniles and adults discarded. So in fact, using the BRD device would save the fishery almost $60 million annually. And this is a significant potential loss when you think about this activity taking place year on year. So what is also interesting is that the BRD and the traditional net had similar marketable bycatch across all trials. and also similar marketable shrimp for 2 of these 3 trials. And in fact, those are the key things that the fishers are looking for. Here we have the t-tests. The t-tests were done, and so we could see it in another way. The mean shrimp weight was equal for the 2017-2019 trials. And in fact, the differences here would be the placement of the BRD device on the what we call BRD net. That's the main difference in the trials. And very importantly, the mean marketable bycatch weight was equal, statistically equal in each of the trials. And it's important for the fishers to have not lost the average marketable bycatch, because this still represents significant income for them. So overall then, it was noteworthy that we had a successful BRD trial where the bycatch was reduced by 47%. But what we noted was that most of the losses that still continue to take place, particularly in traditional net, are from discarded juveniles, not so much discarded adults. And what this and many of the juveniles that are being discarded are juveniles of marketable bycatch. So in fact, you're throwing away juveniles for species that are very important to the market now and in the foreseeable future. So this really means that while the fishers are focusing on higher current profits, in fact, they're losing a lot of potential income by discarding all of these juveniles. So this allows us to have a better idea of the value of what is lost. And this study, I want to acknowledge, was done based on FAO funding under the Sustainable Management of Bycatch in Latin America and Caribbean Trout Fisheries, the RIBIC 2 LAC project. I want to particularly thank Imran Khan, who undertook the BRD trials, who collected data, and so the data for this study is based on secondary data from that study. And I also want to thank staff at the Trinidad and Tobago Fisheries Division, in particular, Mrs. Nerissa Lucky, the director, Mrs. Julian Bennett, and Mrs. Laura Ferreira. I wish to thank you for your attention. Have a great day.
Thank you, Sharon. We have time for a couple of questions. I believe that Sharon is online. So any questions from the audience? And Fadila is going to let us know if there's any virtual questions. It was a very interesting presentation. Anyone? Anyone over there? But Sharon, I have a question. I was struck by all the juveniles are cyanids, which, you know, clearly are really important. So it looks to me like by using this method, you know, not only is it better for the fishers because the juveniles aren't being killed, but it's better for the entire ecosystem. So I just thought that was really important, that there's a way to not be catching all those juvenile sea trout and croakers. Anyone else?
Yeah, thank you.
Oh, there you are. Okay, well, I guess we're going to continue along then. I'm gonna have to wait until that comes up, but I do believe that Jade Carver is our next speaker, and she is competing for the student award. And as soon as we get her title up there, I will introduce her. Okay, so Jade is going to be talking about the impacts of artificial reef development.
Good morning, everyone. Like she said, my name is Jade Carver. I'm a master's student at LSU. And today I'll be talking about the impact of an artificial reef deployment on reef fish movement and community assemblages. So I'm sure most of us are familiar with artificial reefs, but these are just man-made structures that are designed to mimic some of the characteristics of natural reefs. Typically, we deploy them with the intent to either increase fisheries yield or to promote the recovery of an overfished species. And artificial reefs accomplish these goals by providing critical habitat in areas that lack benthic structure. We understand that artificial reefs can enhance fish biomass in a region. However, it's less clear where the source of that biomass is from. This is a topic that researchers have long debated. We're trying to figure out exactly how artificial reefs impact living resource production. We've arrived at these two hypotheses. So the attraction hypothesis suggests that fish from the existing fish from the surrounding area are merely attracted to an artificial reef, whereas the production hypothesis suggests that artificial reefs are providing additional habitat that allows for fish populations to grow. And today, most researchers accept that artificial reefs probably lie somewhere on the spectrum between attraction and production. Artificial reefs shown here in red, as well as oil and gas platforms, which are shown in black, have both been widely distributed and deployed throughout the Gulf of Mexico. These oil and gas platforms are large and complex structures that can provide habitat throughout the water column. And they've been found to support rather large fish populations. As of 2016, there were over 4,000 artificial reefs in the Gulf and over 2,000 oil and gas platforms, although some of these have slowly been removed as they've reached the end of their production lifetime. So this study came about because of the CCA's Reef Louisiana project, which was a conservation effort designed to replace removed platforms with artificial reefs. And this gave us a unique opportunity because we knew both when and where an artificial reef was going to be deployed. And so that allowed us to examine the overall response of fish to a new artificial reef. We had two main questions that we were looking at in this study. First of all, how do existing reef fish communities change after an artificial reef is deployed in an area? And the second, how might fish colonize or utilize an artificial reef after it's deployed? So we used two different methods to answer these questions. The first were baited remote underwater video surveys, or BRUVs. And then we used those to look at changes in fish abundance and community assemblages, both at the artificial reef site and the surrounding nearby structure. We also used acoustic telemetry to document colonization or recruitment at the reef habitat from the nearby structures. So our study site was located about 20 kilometers offshore of Timbalier Island in Louisiana. This artificial reef was deployed in October of 2021. And as you can see, there were a few platforms that were in relatively close proximity to the reef. So those three were within 2 kilometers of the reef site and are included in this study. The artificial reef itself was composed of about 40 concrete culvers. Those were deployed in a 100 by 100 meter area. As you can see, that's a sonar survey image that we did after the reef was deployed, just to get an idea of how the structure was oriented there. The depth in this area is about 20 meters, and the max vertical relief of the structure used was 2.5 meters. For our BRUV surveys, we used a drop camera array composed of 2 action cameras that were suspended in the water column. We used GoPro Hero 9s, and the first camera was placed 3 meters above the seafloor, and the second was 9 meters above the seafloor. Both cameras had a small bait pouch located underneath them, as well as a small fin attached to the back to keep them stabilized in the current. We performed 1 survey prior to the artificial reef's deployment. That was just to get an idea of the baseline around the artificial reef site, as well as the platforms in the area. And we performed subsequent surveys seasonally for the following year. So a total of 5 surveys were conducted, and each survey consisted of multiple 5-minute soaks at each of the locations or structure types within the study area. So that included an open bottom or control site that was randomly selected from the area around the artificial reef that lacked structure, included the artificial reef itself, as well as the 3 platforms in the study area. Now for our second method, in order to get an idea of how these fish might be recruiting to an artificial reef, we took advantage of the fact that in this site, there was platforms in close proximity to the artificial reef. And we used acoustic telemetry to examine that. For this, we used red snapper and gray snapper as our study species. And each fish was tagged with a small acoustic transmitter. We used V9s from Vemco. Those transmitters were surgically implanted into the fish, and each transmitter emits a uniquely coded signal every minute to minute and a half that's then picked up and recorded by these acoustic receivers nearby. We placed 1 acoustic receiver at each of the platforms in the study area. And after the artificial reef was deployed, we placed additional receivers there as well. We tagged a total of 40 fish, and that was split evenly between the two species. So you can see here, all of the fish were tagged at the platforms they were caught by hook and line and then tagged and released at the platforms in the study area. And those numbers are the sample sizes for our tagging effort, correlated by species by color. So the red snapper are in red and gray snapper were in blue, and those colors will be used throughout the rest of the figures in this presentation. So these fish were all tagged prior to the artificial reef deployment, and those tags had an estimated battery life of around 300 days. So our last tag detection was in July of this year. And one thing I do want to mention is that about 2 weeks after we finished our tagging, Hurricane Ida passed by close to our study site. That did affect some of the equipment and the results that we had out there. And I'll talk about that more when we get to the results. So for our BREV surveys, once we completed those, we brought the videos back to the lab where they were then analyzed. We identified any observed specimen to the lowest taxonomic level possible, and then created an estimate of relative abundance using the max-n technique. Those counts were then used to compare species richness and abundance across all 3 structure types in our study area, as well as each season that the surveys were performed in. And we did that analysis using ANOVAs. And here are the results from that analysis. So across the top, you can see the mean species richness, and the bottom plots are showing the mean abundance. And those are across time, so by season that the surveys were performed in. And those colors correspond to the different structure types. So the control or the open bottom sites are in gray, the reef is in yellow, and the platforms are in green, and standard error is shown for each of those as well. So what you can see here is obviously that species richness and abundance was the greatest at the platforms, whereas the artificial reef, we didn't see any changes over time really at the reef, and it wasn't significantly different than the control sites. For the second portion of our analysis, we decided to look at red and gray snapper abundance. We only ever observed snapper at the platforms. We didn't really see any at the control sites or the reef. So this analysis is limited to that structure type. But we looked at their abundance and how it changed over depth and season. And this was also done using ANOVAs. And here are those results. So that figure on the left is showing the mean overall snapper abundance by species and over time. And the figures on the right are showing it separated by species and also looking at it across depth. So those darker colored bars are the abundance estimates from the bottom camera, so the deeper camera. And what we saw here was that we did observe much more gray snapper than we did red snapper. And those gray snapper were typically seen higher in the water column. So depth was an important and significant variable for gray snapper. And while it wasn't significant for red snapper, there is a trend there that's also reflected in the gray snapper trends as well. And that's likely to be explained by the presence of hypoxia in this area. So this study site is located in a part of the Gulf that experiences seasonal hypoxia, a large seasonal hypoxic zone. And that typically occurs during the spring and the summer months, and that's reflected here in these graphs. As you can see, the fish starting to increasingly utilize the upper water column during the spring and the summer. For our acoustic telemetry portion of our study, we were mainly looking to see if there was any migration from this tagged fish from the platforms to the artificial reef site after it was deployed. We also looked to see if there was any overall emigration from the study site. And what we found was that during the initial 9 months that the reef has been deployed, we haven't documented any migration of these tagged fish from the platforms to the artificial reef. There could be a number of reasons for this, but it's possible that this is also related to hypoxia. If the artificial reef isn't providing sufficient vertical relief for those fish to be able to escape the hypoxic conditions, the platforms may still be preferable habitat over the artificial reef. As for the emigration portion, this figure on the left is showing the overall number of tagged fish remaining in the study area. We had in July, at the end of the study, there were still 13 fish in the area, and most of the emigration occurred during the first 2 months of the study. And we can see that in a bit more detail on the figure to the right, which is showing the proportion of fish that emigrated during a certain time bin out of the number of fish remaining in the study area. And so those time bins are shown across the x-axis there. And that first bin is a 48-hour period post-tagging. And so you can see during that 48 hours, we had about 20% of our fish leave the study area. That second bin is referring to a 2-month time period that ended in November, and that was after Ida. That was the first time that we were able to get back out to the site after Ida passed through. And at that time, we were replacing some equipment and found that around 60% of the fish that remained post-tagging had then emigrated. After that, after November, those are split into each 2-month time periods, and emigration was relatively rare and low after that point. So no immigration between November and January, and then it stayed around 6 to 7% for the remainder of the study. So just to bring all of this together, again, we saw no evidence of tagged fish migrating to the new artificial reef, and that, as well as the consistent low species richness and abundance at the reef, could be a reflection of poor habitat quality if that reef is not complex enough or isn't providing the sufficient relief for those fish to be able to escape the hypoxic conditions in that area. It's also possible that because this reef was deployed in the late fall, that prevented the recruitment of newly settled fish in 2021 at the reef, and that could be reflected in the low species abundance there. Also, we did want to point out that the study raised awareness to the fact that hurricanes may play an important role in redistributing fish populations and facilitating movement between structures. And then finally, for further work, it could be beneficial for us to examine the effect of different types of artificial reef structures on and how that could affect reef fish movement, as well as community assemblages in areas with similar conditions as this site. And I'd just like to thank the funding agencies for this project, as well as my advisor and everyone who helped with the field work and made that possible. And I'll take any questions at this time.
Thank you, Jade. Perfectly timed. There are 2 minutes for questions. Andre?
Hi, Jade. Nice presentation. I have a question about when comparing the diversity across structures, and those structures are very different in terms of aerial cover. And we know that diversity tends to increase linearly with the size of whatever structure is holding it. Do you think that you should have accounted for the differences in size between structures to compare diversity estimates?
Right, that's something that we could look at in the future. We didn't include that really in the study. These were just a novice, so it was looking at the mean over the site. So hopefully that kind of helped account for any differences there. But no, that could definitely be something we could look at in the future.
My other question is about the methodology. This was baited video cameras?
I'm sorry?
For the methodology for estimate species, you were using videos, and there were some baiting in there as well?
Yeah, it was baited video.
So I wonder, do we know how the different species respond to baiting, and whether that can actually introduce some bias? If you have some species that are really, very more likely to respond to the bait versus the others,
right? That's
comparison
possible.
to species.
I'm not sure about that. I haven't looked into that. Most of the fish seem to respond pretty well. We saw pretty like an even spread of the species that we saw with the bait. So I don't think it influenced it too much, but it could be something to look into.
Thank you.
Thank you.
Kim, I see you have a question. We've got a second, so go right ahead.
Thank you. That was a great presentation. It's quite elaborate to remove a platform and put an artificial reef in, it seems. What do you think about toppling existing reefs? Do you think that would provide better results? Or with your hypoxia results, it seems that the vertical structure was important as well. What's your
I think
thoughts?
I know the CCA has used toppled reefs, or at least the top of the platforms and other reefs that they've been using in this project. This is just what they had available for this reef, and that worked with our timeline. I do think that could probably lead to more abundance and greater diversity at the artificial reef site, definitely.
Great.
Thank you.
Thank you.
So Fidela tells me that we have a virtual question, so we're going to give you an extra tech question. Go ahead, Alfonso.
Good morning. I didn't know I was going to talk live. Good morning, everyone. I'm curious, why didn't you measure the environmental variables at the sites? Why didn't you measure them? Because you measured that you mentioned that the hypoxia is a possibility that is influencing the abundance. But why didn't you measure that?
It just wasn't something that we were really aware of, I think, at the beginning of the study, and that became more clear as we went on. We're working on a different portion of the study right now that is looking at hypoxia and the reaction of these fish to that. So we are currently measuring it. It just wasn't done at the time that this study was happening.
Okay, another question. Why did you include the hurricane's influence in your conclusions? Because it is not part of your work. I mean, it's just suppose. I mean, it's not part of your work. Why did you include that hurricanes is probably an influence of abundance?
Right. We thought it was an interesting result that we saw there. Our data wasn't complete because we had some lost equipment, but it passed by relatively close to my study site. And so that's not something that happens every day. And so we thought it was important to include that was an effect that we did see here.
Okay, thank you. I see
Yeah,
it.
yeah, but it's not part of your conclusions. Thank you.
So I'm going to have to cut it off here. Apparently, everyone is very interested. Rich, you'll have to catch Jade at the break. I'm sorry. Okay, our next presenter is Jamie Clark, and she's going to be talking about aging fish with Fourier transformation, infrared microscope, whatever. You guys can read it.
It's a mouthful. Is it? All right. Good morning, everyone. I'm an implant in this session, by the way, if you can't tell. My name is Jamie Clark. I'm a NOAA affiliate with the University of Miami's CMIS, which is the Cooperative Institute for Marine and Atmospheric Sciences at the NOAA Beaufort Lab. I'm here to talk today about predictive aging of fish with Fourier transform near infrared spectroscopy. It's a mouthful. And we have been doing this with two different genetically distinct stocks of white grunt in the waters of the US South Atlantic. So not the Gulf or Caribbean. Is it? What is it? Oh, there you go. Oh, there it is. Okay. Oh, there we go. Okay. So briefly, I'll go over traditional aging of fish, then FT near spectroscopy background, theory. I'll go into our project objectives, methods, outcomes, and future directions, and then I'll leave you with some key takeaways. So the Biology and Life History Group at the NOAA Fisheries receives tens of thousands of aging structures per year from fishery independent surveys and dependent port samplers. These structures vary between otoliths, spines, vertebrae, and scales from lots of different species of commercially important fish. Each type of, there we go, oh, sorry. Each type of structure requires specific processing method. Some are read whole if validated, but otherwise we section the majority of structures on either the Hillquist or isomet. If read whole, we do section them on the isomet and then read them on the microscope afterwards. Oh my goodness, I'm all over the place. Okay, so calculating fish age can be difficult, species dependent, requires training, reading consistency, and institutional knowledge over time. It's difficult to consistently age fish over a specific length of time, just because it's difficult to keep a consistent reader at the lab. We've had high turnover, so it's been difficult to train personnel. And throughout this entire aging process, we do perform QA/QC on all associated data to identify any early onset outliers or data gaps that require our attention. Fourier transform near infrared spectroscopy, it's a mouthful, I'm going to call it near spectroscopy in this presentation, is a non-destructive method that passes near infrared light through a sample which excites chemical bonds and forms a spectral signature of absorbance measurements at specific wavelengths seen in the above image. This spectral signature quantifies the concentration and presence of organic bonds in the studied sample. So you can compare the chemical composition to the spectral output. These certain peaks correlate to certain bonds in the sample, so you can sort of correlate to what actual compound is in that sample. Near-infrared spectroscopy has been used in the agricultural and pharmaceutical industries since the 80s to measure butterfat percentage in milk or even drug purity in prescription drugs, among many other applications. More recently, though, as you heard on Tuesday's talk by Beverly Barnett, near-infrared spectroscopy has been successfully used for the age determination in the otoliths of red snapper and walleye pollock, as well as the vertebrae of sharks and rays. This is done by comparing the spectral signatures of known age structures to unknown age structures via regression modeling. In this plot above, average otolith absorbance increases with increasing fish age. So this is average spectral signature per age class. Eventually, FT near spectroscopy age prediction data can be contributed to the upcoming white grunt stock assessment, aging roughly 30,000 white grunt otoliths more efficiently than traditional aging. So with NOAA's strategic initiative and MARFIN funding, our objectives are to first develop standardized practices for FT near spectroscopy sampling across various NOAA labs. Second, to create 2 different cross-validated calibration partial least square models for both the Carolinas and Florida stocks of white grunt. And third, apply this calibration model to a test validation set to assess prediction accuracy by predicting unknown aged samples. So originally, the South Atlantic white grunt stock assessment was planned for 2023, but has since been pushed back to 2027. The Beaufort lab had about 32,000 otoliths to process and age for the stock assessment. So white grunt voted well for this type of analysis. For this study, I weighed, scanned on a Tango spectrometer, processed on the Hillquist, and aged 1,390 otoliths from both the Carolinas population and the Florida population. The literature had identified 2 genetically distinct stocks of white grunt in the waters of the US South Atlantic, the Carolinas and Florida. So as the morphometric data will show, these genetic stocks have different growth characteristics and therefore should estimate age, predict age in 2 different models. Fish labeled as Carolina stock were landed in North Carolina or South Carolina, and fish labeled as Florida stock were fish landed in the Atlantic coast of Florida. I performed QA/QC on the data and created 2 different preliminary calibration PLS models. The next step this winter will be to identify or define each stock's calibration set, then apply the remaining spectra as a test validation to each stock. So looking at the morphometric data and the need to separate the 2 stocks, here are size at age plots of the Carolinas and Florida. The Carolinas is and will stay on the left, and the Florida is and will stay on the right. Fork length is in millimeters on the y-axis, and calendar age is in years on the x-axis. Calendar age is calculated by taking the month that the fish was caught into consideration when assigning it to an age class. The Carolina fish were larger at age and contained older ages than Florida fish. You can see that there's a bit of a data gap in the 17 to 23-year-old age range in the Florida population. An additional morphometric is otolith weight, which has been found to be proportional to age. Here, otolith weight in grams is on the y-axis and calendar age in years is on the x-axis. Sorry. So again, this is otolith weight. Otolith weight is on the y-axis in grams and calendar age is on the x in years. The otoliths of the Carolina fish were heavier at age than the Florida fish. Since otolith absorbance for near infrared spectroscopy correlates with higher mass, this further confirms the necessity to separate these two stocks into two separate PLS models. So from here on out in the presentation, I'll be using fractional age. Fractional age will be used because it's a more accurate estimate of how long that fish has been alive. It's sort of a decimal age of the length of time. And this takes into account peak spawn, date of capture, and calendar age into that. The peak spawn was found to be different per stock in the literature by 1 month. And those fractional age calculations are stock specific. In general, you can see that we see an increased otolith absorbance with increasing fish age. It's more prominent in the Carolina population because we had older ages in that population. So you can see the very distinct red, the older otoliths at the very top of the Carolina spectra. This is just a preliminary look at the data, and spectra have not been averaged per year class. For the Carolinas partial least squares model, the assigned fractional age is on the x-axis and the predicted is on the y. Ideally, there would be a 1-to-1 relationship. So that would be 100% agreement between my reads and the FT-near spectroscopy estimates. The R squared here is 90.57, and the root mean square error of cross-validation is 1.13, which means the model predictions are within 1.13 years of my reference age. This is generally improved from traditional aging, which can range between 1 to 5 years of error. The RPD here is the residual percentage difference is over 3, which indicates that it's a good fitting model. This model is mostly ready for a test validation set this winter, but I'll show you the Florida next, which it needs some work. So this is the Florida partial least squares model. The axes are the same as the previous slide, where reference is on the X and predicted age is on the Y. The R squared is 86.64 here, and the RPD is less than the optimal 3. So I do have a ton of samples in this calibration set, and I do plan to parse them out this winter and work a bit more on this. I believe that this model has a bit more of a difficult time predicting age because of that plateaued relationship between otolith weight and age that you saw in the Florida fish. Since otolith weight is very correlated with spectral output, they needed to be modeled differently, and it's hard. Yeah, the otolith weight is pretty much similar between ages 5 to 13. Optimistically, I would like to add 0 to 1 and 13-plus-year-old otoliths to the calibration models of both stocks. I also hope to adopt a second age reader to ensure our reference ages are as accurate as we can calculate. Over the following winter, I will work with data scientists to tease out my calibration versus test set for each stock to ensure they are geographically and temporally representative. We'll also work with partners to develop our PLS models in R rather than Opus. And we will also create a spectral database to associate spectral files to metadata such as age, otolith weight, and county landed. So in overall, FT-near spectroscopy is non-destructive to the aging structure. It's more efficient than traditional aging when those calibration models are built. And we observed good correlation between 1, otolith absorbance and age assignment, and 2, reference age and predicted age. We also found the Carolina fish growing to larger sizes, older ages, and possessed heavier otoliths than the Florida fish. The Carolinas model predicted age within 1.13 years of the reference age, and it had favorable RPD scores, so we'll be going forward with the Carolina model. The Florida model still needs some work, so we're going to work on that this winter and make sure it's representative geographically and temporally. Although this method can optimize production aging, we still have to continue traditional aging over time for precision and continue subsampling those samples, future received samples, to add back into those models to ensure that those newer years are included in each model. The end goal is to provide size at age data for stock assessments with accurate age predictions derived from FT-near spectroscopy age predictions. So with that, I'd like to thank all of my collaborators from NOAA, especially Andy Ostrowski and Dr. Beverly Barnett, who you heard earlier. My talk was supposed to be Friday, so I wanted to thank all the veterans for serving, and happy Veterans Day. Thank you so much for listening to Predictive Aging about on white grunt with Fourier transform near infrared spectroscopy. It's a mouthful, so you don't have to say it. I'll say it. Thank you.
Thank you, Jane. Nice job. And we have plenty of times for questions, if anybody has questions for Jamie. Okay, there's a virtual one. Do you want to read it or can they just talk it?
This one is from Ron Hill. I don't know where he
Where
is.
is Ron?
He asks, is there a clear geographic break between the Carolina and Florida stocks? Could a fuzzy boundary between the two account for some of the poor fit for Florida?
Ooh, great question, Ron. So that is a great question, and we're going to be investigating that. Currently, we have just been going by county or pretty much state landed. And so right now I do see those larger fork lengths fish in the Florida model. So those are potentially maybe a hybrid of the Carolina and Florida populations. So there's definitely going to be some genetic testing, hopefully in the future, to ensure that there's distinction between those two stocks.
Anyone else? No? Okay, well, thank you, Jamie.
Thank you.
Mhm. Okay, I've got to wait. Okay, so our next talk is by Anna Oswolski. Did I just totally destroy that? And she's going to be talking about characterization of depredation in the Gulf of Mexico reef fish fishery.
Hi, everyone. My name is Anna Osowski, and I work with the Marine Fisheries Ecology Program at Mississippi State University, and I'm excited to be here today to discuss some work we have been doing with regards to depredation in the Gulf of Mexico. Depredation, defined as the partial or complete removal of a hooked fish by a non-target species, has been a topic of significant concern worldwide and has been escalating in frequency in the Gulf of Mexico. In recent years, shark depredation interactions in particular have been increasing, mirroring increases in the numbers of fishers on the water, as well as some recovering shark populations following decades of overfishing. These interactions have substantial economic consequences for commercial and recreational fishermen. Fishermen must contend with increased handling time, extensive gear repair, and shifting fishing locations in order to decrease or at least minimize these interactions. Additionally, these interactions have negative effects on the accuracy of fisheries assessments as well as fisheries management efforts. Although fishermen have urged fisheries managers to address these increasingly prevalent interactions, a comprehensive understanding of the nature, extent, and magnitude of depredation in the Gulf of Mexico is currently lacking. Therefore, we work to co-produce a shared characterization of depredation in the Gulf of Mexico reef fish fishery as a necessary precursor to quantify depredation and to begin to develop effective solutions. Co-producing a shared characterization of depredation in the Gulf of Mexico reef fish fishery was the result of 3 phases. First, we worked to gather, analyze, and interpret fisheries data from the National Marine Fisheries Service Southeast Fisheries Science Center Reef Fish Observer Program to characterize depredation in the Gulf of Mexico. The bulk of this work is summarized in a paper by Duffin et al., but I'm just going to go through a couple of results that we found. Bottom longline fisheries data and vertical longline fisheries data have demonstrated increased cases of depredation shown in red on the graphs to the right, and shark presence shown in blue was a good predictor of depredation. Once we gathered existing information on depredation in the Gulf of Mexico, we designed and implemented an electronic survey that was depredation focused to 1,000 Gulf of Mexico recreational and commercial fishermen. This survey was designed to measure participants' ecological knowledge of depredation, as well as their attitudes towards depredation. Survey responses were then incorporated into fuzzy logic cognitive mental models that were then subsequently combined to create community models like the one shown on the right that capture group beliefs and perceptions on depredation. Once we gathered existing information from phase 1 and then created community models from phase 2, this laid the groundwork for phase 3 of the project to design and implement a collaborative workshop with the goal of co-producing a shared characterization of depredation in the Gulf of Mexico reef fish fishery. This third and final phase will be the focus of my talk today. Our workshop was convened in April earlier this year and had 3 primary objectives. First, to facilitate in-person discussion and reciprocal learning between researchers, resource managers, and stakeholders. Second, to develop, assess, discuss, or to allow stakeholders to develop, assess, discuss, and refine Gulf of Mexico reef fish depredation community models. And finally, to identify knowledge gaps in our understanding of Gulf of Mexico reef fish depredation. More than 20 charter for hire captains from all 5 Gulf of Mexico states contributed to a participatory mapping process during this workshop. Participatory mapping is a research method that allows stakeholders to shape discussion and share viewpoints with minimal intervention from researchers. First, stakeholders were divided into breakout groups and were presented with 4 core model concepts that were identified from the survey in phase 2 to be important to understanding reef fish depredation in the Gulf of Mexico. These 4 core model concepts are illustrated on the left-hand side of the slide and include reef fish populations, shark populations, angler satisfaction, and effective fisheries management. Additionally, we also provided a word bank of 20 additional concepts also taken from the survey in phase 2. And those are shown on the right-hand side of this graph that encouraged in-depth and thorough discussion during those breakout groups. Stakeholders were encouraged to participate in active discussion and to incorporate concepts, either from the word bank or on their own in the form of post-it notes based upon their personal knowledge and experience. Discussion between researchers and stakeholders also revealed underlying relationships between these components and how changes in these components would result from changes in perceptions, actions, and environmental processes. Stakeholders were asked to assign categorical weights to these relationships in the form of a negative or a positive number to correlate with the effect that changes in one component would have on another. And these are illustrated in the red and blue lines that are shown in that photo on the right-hand side. These concepts and relationships were then incorporated from post-it notes into a final community model like the one shown here. Components that were considered important to understanding Gulf of Mexico reef fish depredation are shown in boxes, and they're connected by those red and blue lines that indicate what type of relationship changes in one component would have on another. Because we were able to develop these community models, we were able to reveal distinct differences in the extent of depredation, as well as perceptions of depredation for each Gulf of Mexico state. We were also able to evaluate and identify our knowledge gaps in our understanding of Gulf of Mexico reef fish depredation, particularly as it relates to developing effective solutions. Because effective fisheries management and shark populations were 2 of those 4 core model concepts that were identified to be important in breakout group discussion compared to other components presented, these were incorporated in scenario analyses. These analyses allow us to determine how each community model would be altered by changing components of interest. For these analyses, we increased effective fisheries management and decreased shark populations to determine how each community model would change. Because each Gulf state had a unique community model, this resulted in different outcomes for each Gulf state. For example, the Florida community model, these changes resulted in relative increases in coastal heritage, relative increases in ecosystem health, and relative decreases in shark depredation. Stakeholder breakout groups also contributed to participatory mapping exercises to determine temporal and spatial depredation trends. Although some stakeholders attributed the rise in depredation interactions to increased shark populations, there was no consensus on which shark species were predominantly responsible. Ridgeback shark species, such as dusky sharks, silky sharks, and sandbar sharks, as well as bull sharks, were considered to be primary depredating species, along with dolphins. Goliath grouper, as well as greater amberjack, were also considered to be common depredator species, although these species were not discussed in detail. Stakeholders also were able to identify areas of high numbers of depredating species that seem to overlap with areas of high fishing pressure. While most stakeholders agreed that instances of depredation have been increasing, there was division between stakeholders as to whether or not shark populations have been increasing or decreasing in recent years, and whether these increasing instances of depredation are the result of fisheries management efforts. A further discussion between researchers and stakeholders revealed a general mistrust between stakeholders and fisheries managers. Past efforts to protect declining shark populations have anecdotally now resulted in an overabundance of shark species that skews the ecosystem balance upon which stakeholders depend on for their livelihoods. A directed and expanded shark harvest was presented as a viable solution to decrease these interactions. However, stakeholders recognize two primary barriers to the establishment of an expanded and directed shark harvest. First, a polarizing public opinion on shark harvest that is somewhat the result of past conservation efforts. Specifically, messages supporting shark conservation have resulted in a perspective that even if shark populations are healthy, there continues to be a need for very stringent shark conservation practices. These past messages, as well as the public opinion that upholds this perspective, have effectively prevented stakeholders from using sharks as resources. Secondly, a lack of infrastructure as well as demand for a market for sustainable shark products. Stakeholders contended that fisheries management efforts must address current regulations that effectively prevent the long-term establishment of an expanded shark harvest. For example, Florida currently has a ban on shark fins. Texas has a commercial shark harvest ban, and current federal regulations prevent Mississippi stakeholders from reaching the blacktip shark quota. Other stakeholders during the workshop recognize that changing the tide of public opinion, as well as going through these management barriers, may not be feasible, and they determined that effective shark deterrence may represent a better solution to addressing depredation. To conclude, depredation is a complex sociological, ecological issue that requires innovative, multifaceted solutions. The community models that we developed allowed us to evaluate, to an extent, which solutions may be most effective to mitigating depredation. Excuse me. Additionally, the co-production of knowledge that was fostered during this workshop served several valuable purposes. First, we were able to tap into a wealth of local ecological knowledge. Second, this enabled us to further characterize depredation, as well as identify knowledge gaps that can be addressed through further research questions. Third, stakeholder knowledge and beliefs captured during community models reveal a disconnect between species conservation efforts as well as ecosystem balance, resulting in a mistrust between stakeholders and fisheries management. And lastly, even though there was this mistrust, our workshop fostered trust between these entities as these three entities came together to better understand depredation in the Gulf of Mexico reef fish fishery. Funding for this project was provided by NOAA's Restore Science Program under its award to Mississippi State University. Thank you for your attention. At this time, I'd be happy to answer any questions. Thank you so much.
Thank you, Anna. Good job. And we have plenty of time for questions. If anyone has questions, Rich?
Yeah, a question and a comment. First of all, it was a very nice presentation. So on your geographical distribution, you had a very different scenario for the Florida and areas more west, which I'm not from the Gulf, but I think that's all where a lot of the oil platforms and things are. And would there be a higher predator population around those things that could drive that higher rate, or is it just great? Because you mentioned greater rate of fishing pressure, which of course obviously impacts it as well.
That's actually a really great question, and I don't know the answer off the top of my head, but it's certainly something that we can incorporate into future analyses.
Okay, and my comment was the way you were presenting this, you were separating stakeholders to like not include the general public who would be represented mostly by the conservation NGOs. And I think general public has a role to play because it is a national resource. And so I would consider them part of the stakeholders.
Absolutely.
Thank you. We've got time for one more question.
Hi, Anna. Great talk.
Thank you.
And thank you, Kim from Marine Lab. I was wondering if you could reflect a little bit more on when you brought the fishing stakeholders together. You mentioned the deterrence, and I was wondering if there was anything that they had put out there that was being effective at this time for both sharks and dolphins, and reflect on it. Or is this report going to come out that's going to have some of those so it could be more broadly shared? Thank you.
Absolutely. The report will have more details, and that was kind of mixed between stakeholder groups. Overall, some were, excuse me, some were effective and some were not effective. And then there were other ideas that there wasn't a lot of information on, but that future research efforts can focus on evaluating the efficacy of various shark deterrents out there.
Okay, thank you. Nice job.
Thank you so much.
And I believe this is this our final presentation for this, or do we add another one? Is this our last one for this session? One more? Okay, but we've got like multiple ones. So your name is not up there, so I'm gonna have to read your name tag. Oh, Kara, I'm sorry. Okay, so Kara's going to be talking about effects of habitat and survival and residency of red snapper. Yeah.
All right, so I'm going to present today some preliminary results from a study we just wrapped up about 2 weeks ago, monitoring red snapper movement across multiple habitat types on the West Florida Shelf. I'm gonna go forward. So I work in a lab that uses acoustic telemetry to track. Oh, my bad. This is the problem when you do these things. Sorry, guys. Okay, here we are. I work in a lab that uses acoustic telemetry to track fish throughout the Gulf of Mexico. So we surgically implant our fish. You can see Hayden making an incision on this fish up here. And on this map shows the locations of all the places we are currently tracking our tagged fish in the Gulf. So this study takes place in this gold box you can see up here on that map. And so this array was located about 80 kilometers offshore of Tampa Bay, and it was in about 38 meters of water. So it consisted of 67 receivers and was designed as a VPS. So our receivers were actually close enough together to triangulate the exact location of the fish as they move about. And for this study, we were interested in examining how habitat is affecting red snapper movement, but specifically during the fishing and spawning seasons, both of which co-occur on Florida's Gulf Coast from June to October. We're interested in this season because this is when the fish are going to be exposed to a sudden increase in fishing pressure as the recreational season opens on June 1st, and because the spawning season may be increasing their mobility. So essentially, during this time of year, when we're anticipating to see the highest rates of mortality, as well as the possibly higher rates of immigration. But first, we needed to know what kind of habitat was out there, right? So we asked our colleagues over at the state's Fisheries Independent Monitoring Program to actually go out and side scan sonar map our entire study area. And what we saw is that there are 3 different habitat types within the boundaries of our array. The bulk of the habitat was this low relief contiguous hard bottom. It's what you see all of that blue is. But we also had one teeny tiny wreck, which is that green dot up there, as well as a very small ledge, which is that red dot. And both the artificial reef and the ledge were fairly small structures that were isolated by the nearest hard bottom by almost 1 kilometer of sand in every direction. Just to give you an idea of kind of what they look like, this is the side scan image on the left, and this is a picture from our camera surveys on the right. The light color in the side scan is the hard bottom, and the dark color is the soft bottom. So flat hard bottom is pretty boring. It's very flat reef, and you almost only know that there's rock there because there's sponges and things growing on it. Most of the time it's covered in a little layer of sand. Not a lot of complexity to that bottom. The ledge was, you can kind of see it, there's a little like horseshoe, the idea of a horseshoe shape in that scan. And this was our like high relief natural bottom. And in the Western Florida, the West Florida shelf, that sometimes can mean like a 1/2 meter relief. So that's what we had here. And then our artificial reef, you can see, was 2 barges that sunk sitting next to one another. So they're pretty old and beaten down. They're not a publicly sunk number, so it was an actual wreck, but it's fairly known in the fishing community. And again, not very big, but there's about a 1/2 meter of relief on these sites. So having these 3 habitat types is nice, because up until recently, there hasn't been a ton of red snapper work done on natural bottoms, especially this like large areas of contiguous bottom. So we can now look at how our fish are surviving and the movement on the natural bottoms and compare it to our wreck, which will kind of ground it in the greater context of the existing snapper telemetry research. In order to examine these habitat effects, we wanted to make sure that the fish we were tracking were actually like resident individuals. For those of us who track fish, we know that there can be a lot of individual variability in fish movement. So here are 2 fish that we tagged on the same site at the same day, the one in blue and the one in white. You can see the one in blue is using much more space than the one in white. So oftentimes this is independent of size and sex. It's just like some fish really like to move more than others. And so to try and control for any like natural proclivity that these fish might be having to readily leave the array right after we tag them, we actually tagged all of our fish in the winter. So we tagged them in the winter. We wanted to observe them in the summer. So we had 6 months of tracking data prior to June 1st to ensure that the fish we were looking at were actually relatively resident individuals. We tagged a total of 60 fish between 2020 and 2022 and had pretty good survival. We only had 7 mortalities. They were all due to discard predations. And ultimately had 46 individuals in the array. So they stayed all spring and all winter, and then they were there on June 1st to start being tracked. And we ended this tracking period after 140 days because we pulled this array in mid-October and we wanted it to be standardized. So some results. What we saw was that survival and thus residency was actually lower on our rec site than on our natural hard bottom. So this bar graph shows the proportion of our fish that actually made it through that like 140-day gauntlet in our array with the average tracking times listed above. And it's worth mentioning that the fish we tagged on the wreck, they stayed there the whole time, all of them. So they were there when we tagged them in the winter, they stayed through the winter and the spring. And then when recreational fishing season opened on June 1st, 2 of them immediately died. And within the first 20 days of June, we lost 80% of our fish on the wreck. So it's pretty notable, but that trend of like the mass mortality or mass disappearances early in June was pretty typical to what we saw throughout the whole array. Of the fish that actually left for the 140 days were up, 2/3 of them did so in the month of June. But in general, the fish tagged on the natural habitat had greater survival and longer tracking times than the wreck fish. So the real question is what might be driving these differences in survival? We know there's a lot of work that's been done on red snapper mortality and fidelity, and we see large differences sometimes in fate, just depending on where the tagging location is. So for us, we were wondering, what is it about this wreck that makes it such a seemingly risky place to live? So to try and better understand this, I looked at the little bit of VPS data we've gotten back so far. Unfortunately, I don't have data for the wreck and the ledge. I only had fish that were tagged on the hard bottom. But what I did see in looking at those fish is that they're displaying a very strong aversion to spending time over sand bottom. So over 80% of the VPS positions that our fish were placed did so over hard bottom habitat. And I think it's really the visuals that drive this point home. So I'm going to show you some maps of our individual fish movement, starting with this area in the black box. And remember, the light color is the hard bottom and the soft color is the sand. So we see that time and again, they are really hugging the contours of these hard bottom habitats and not spending a lot of time out over this exposed sand area. We see them move up, this guy moved up to some of this stuff to the north and to the west, but they don't really ever get detected in this exposed sand. And again, for this larger area, we see I just think it's so cool, right? They're moving around the edges of these reefs and not really going out in the sand. And then we can see the individual variability. This one really liked to go out to this little island of rock. And so, unfortunately, with the way this study was designed, we don't have the ability to know why these fish are avoiding sand bottom, but we can imagine that it's probably a little bit more risky for a fish to venture out over large expanses of sand, right? As they move away from hard structure and cover, it's going to expose them to more risks. So thinking in the context of our wreck and our ledge site, both of which are small, isolated features, right? They're surrounded by almost 1 kilometer of sand in every direction. When a fish wants to seek sanctuary on hard structure, for some reason, they're going to have to stack up in these higher densities, possibly exposing them to increased fishing pressure and predators. And so to wrap it all up, again, these are preliminary results, right? We just pulled this array 2 weeks ago. So I don't have my second round of VPS data back yet. But I'm really excited to look at it and see what the fish are doing on our wreck and our ledge site and see if these trends hold true. Because what we've seen so far is that time and again, when these fish are at their most vulnerable, either because they're exposed to an increase in fishing pressure or because they're moving more due to spawning behavior, we're seeing relatively greater mortality of red snapper on the artificial structure compared to the natural sites. And the bulk of these fish are disappearing early on in the season. And this might be because if they're living on these large hard bottom features, they have the ability to disperse more without having to move over the sand, which we've seen in other studies. When people look at fish movement on large artificial reefs, these fish have access to more nearby structures and their home ranges are larger than when tagged on isolated structures. I think this is notable because this flat hard bottom habitat is what is most readily available to fish, the red snapper on the West Florida shelf. So this habitat type makes up 50% of the available hard bottom to our fish out here, which is very different from what we see in the other parts of the Gulf. Artificial reefs really only make up less than 1% of the available habitat to fish on the West Florida shelf. So knowing how these fish are moving over these contiguous natural bottoms are going to help us continue to improve our management of the stock, especially as ranges continue to expand through adult dispersal. And that's it.
Thank you, Kara. There's plenty of time for questions. Go ahead. Is that Bryce? Okay, I couldn't see you back there.
Hi, awesome talk. I'm wondering whether or not you have the resolution in the data to see where apparent natural mortality effects happen, like predation, based on the VPS data. And then also if you're seeing in the acoustics, can you see a signature of what appears to be, the tag in the belly of a predator?
Yeah, we do. So I bulked everything together as fish that were left, either immigrating, harvested, or predated. But we definitely can see, you can tell, right? When a fish gets eaten, it's just zipping around the ray, and all of a sudden it's like, yeah, that's not a red snapper swimming at 1 meter, moving miles and miles in a day. So we had a fair number of mortalities at the wreck were actually tags that just died. So they were moving around for like 6 months, and all of a sudden they fell out, probably due to a predator biting that fish in half, right? We internally tag them, and the tag stays in when we suture them closed, it's all healed up. But
Other questions? Go ahead.
Hi, Karen. Do you see any effect based on the different sizes? Are big fish acting different than the smaller ones?
so most of our fish were in a fairly small size range, so they were somewhere between like 15 and 24 inches. We didn't have any like big ones, which is a bummer because they probably are using the space a little bit differently. But I know other studies have shown differences in them. We just haven't seen that.
Anyone else? If not, I've got a question. So, and I might have missed this, but was the depth of your hard bottoms the same as of your ledge and your artificial reef?
Yeah.
So, and do you have video to show the density of the fish at the artificial structures versus I'm guessing that there's a whole lot more fish crowded around the structure.
Yeah, so actually our ledge site had the most fish. So it was, isolated, and I think a lot fewer people fish it. So the red snapper density on that ledge was just through the roof, right? We could like chum them to the surface. It was really cool. And then the wreck was the next, and then the low relief hard bottom habitat, really low densities of fish, but they were just there swimming.
Okay, thank you. If there's not any other questions, I guess we'll continue on. There's no virtual. Okay, thank you very much. Okay, and our final talk of the day is Emily Waddell, and she's going to be talking about anthropogenic noise on larval fish behavior. So I think we finally have a pelagic sort of talk here.
Sort of. Okay, good morning, everyone. Thank you for sticking around, and I promise to be as animated and enthusiastic as possible before this coffee break. It's much needed. All right, so let's just dive right in. So we all know that it's important for an individual to find a suitable habitat because it affects their survival and fitness, which ultimately then affects the population's success and persistence. Now, there are many marine organisms that can use various cues to find these habitats, such as physical, chemical, auditory. And for example, there are species of fiddler crab megalope that will settle and molt faster in the presence of conspecific adult chemical cues than odorless seawater. Additionally, if we look at the figure in the bottom right, we can see that northern rock sole larvae will settle slower in gravel sediment because it is larger size than their preferred habitat, which is sand. This talk, however, will focus on auditory cues. So it's super important to look at acoustic cues because sound propagates farther and faster in marine habitats and waters than in air. So auditory cues are used by larval and post-larval forms of many marine organisms, such as crabs, oysters, lobsters, coral, and fish. And these organisms will use these acoustic cues to orient, settle, and metamorphose. And sound cues, especially ambient, are very important to look at because they can relay information about the health of the environment. For example, Gordon et al. Was able to study and look at the soundscape, so all the sounds in the environment of a reef, pre and post a hurricane. And they noted that the acoustic soundscape changed after the hurricane. And they noticed that there was less acoustic richness and the overall sound was quieter post-degradation, which then led to less juvenile fish settlement. Therefore, it's important to study which animals use ambient cues to settle and orient, because the world's oceans are becoming noisier due to human presence. So as we can see in the figure on the left, we have frequency on the x-axis, and then the hearing range of various marine organisms, and then the frequency range of sound produced by various anthropogenic activities. And we can see that the hearing range of fish, which is commonly below 1,000 hertz, nicely overlaps with the sound frequency produced by all of the anthropogenic noises, such as dredging, seismic surveys, and ship propeller noise. Therefore, it's predicted and expected to see that many anthropogenic noises will disrupt and delay larval behavior. So if we look at the figure on the bottom right, we can see that cardinal fish larvae, when exposed to just reef sound, they would spend more time near the speaker when just reef noise was playing. However, when boat noise was introduced to reef sound, less larvae spent time near the speaker. So anthropogenic noise decreased the amount of time spent near the speaker, causing the larvae to become disoriented. This then led to the purpose of my study, which looked at the effect of anthropogenic cues and ambient cues on larval estuarine fish orientation. And I wanted to see if the 4 estuarine species in my study used environmental acoustic cues to orient, and then how anthropogenic noises impacted that orientation. So the 4 species in my study were Florida blenny, southern flounder, red drum, and spotted sea trout. Florida blenny were chosen because they're a small, important cryptobenthic species in oyster habitats, but they settle in seagrasses. And then the latter 3 species were chosen because they're very ecologically and economically important in the Gulf of Mexico as a sport fish. The middle set of images are the settlement-sized larvae used in my study. As you can see, they're adorable, and they ranged in size from about 4 to 8 millimeters, depending on the species. 25 larvae of each species were exposed to one of my 4 sound treatments, which were control, ambient, which was seagrass beds, air guns, and a large ship propeller noise. So all of my trials were tank-based experiments. If we look at the figure on the right, you can see the experimental setup with a clear PVC tube, which acted as the environmental acoustic chamber, and it was placed at the surface in between 2 speakers. The speakers were each hooked up to their own amplifier, and then one amplifier was connected to the computer, which contained the sound files. Prior to conducting my experiment, I had to look at the sound field within the tube because I wanted to see a gradient, obviously with there being louder noise near the actively playing speaker and getting quieter throughout the tube. And I was interested in looking at the sound field measurements between 0 and 2400 hertz, since that's the hearing range of most fishes. And the recording locations are indicated by the X's. So all of these experiments were conducted at night after sunset because most larval activity occurs then. Each night, the actively playing speaker was alternated so that 25 individuals of each species was exposed to sound from the right, and then half was exposed to sound from the left. So each night, each larva was exposed to one of the 4 sound treatments, which was randomly selected. And it was placed in the acclimation tube in the middle of the acoustic chamber for 5 minutes. And then it was exposed to one of the 4 treatments for 6 minutes. And then throughout the duration of the 6-minute trial, the larval position was tracked in relation to the actively playing speaker. So to determine if there was preference or avoidance for each sound, I used a paired Wilcoxon-Steinbranch test. And this was done by comparing the time spent near and away from the actively playing speaker. So the time spent in the 4 ninths closest to the actively playing speaker were summed to create the time total spent near. And then the 4 ninths away from the actively speaker were summed and equaled the time spent away. We also wanted to see if habituation occurred during this short 6-minute duration trial. So to do that, we just summed the time spent near and away for the first minute, as well as the first 3 minutes. So moving on to results, we did notice a good sound gradient within the tube. So this table shows the difference in sound pressure levels between the near and middle location and between the middle and far location. For those of you who aren't as familiar with acoustics, even though it looks like the sound pressure level difference was between 2 and 6 decibels, that's actually a fair amount difference. So looking at the behavioral responses for just the 1 minute duration. So on the x-axis for all of these figures, we have the sound treatment. And on the y-axis, we have the time in seconds spent either near or away from the speaker. And as we can see, the 3 latter species, so southern flounder, red drum, and spotted sea trout, all spent significantly less time near the actively playing speaker when airgun noise was playing. And then if we were to look at their behavior at a 3-minute duration, we see that for the southern flounder larvae, they continue to spend significantly less time near the actively playing speaker when air guns were playing. However, this trend was not continued for the other species. And in fact, for spotted sea trout, we noticed significantly less time spent near the speaker when ship noise was playing. And then interestingly, there was no significant difference spent near or away from the actively playing speaker for the full duration 6-minute trial. And none of the species showed a preference for their ambient sound. So how do our observations compare to others? They don't. So if we remember back to the intro, the majority of other studies noticed that there was an attraction to their ambient sound cues and an avoidance to anthropogenic noises. However, in this study, we noticed that there was no significant response to their ambient soundscape, and there was only an initial avoidance to anthropogenic noise, specifically air guns. However, there was one study conducted by Simpson et al. In 2010 that showed behavioral plasticity in pomocentrid larvae. So in their study, their larvae were exposed to 3 different sound treatments during rearing. It was either reef, so their ambient sound, a pure tonal sound, and then silence. And then the larvae were exposed to the 3 different sound treatments. And they noticed that larvae that were reared listening to reef sound cues spent more time towards the speaker. However, larvae that were reared in silent treatments, like my larvae, when they were played back reef sound cues, they did not spend a significantly different amount of time near or away from the speaker. So one reason that I think we didn't see what others saw is because the majority of other studies looked at reef species and one looked at a pelagic species. Whereas in my study, we had 4 estuarine species, which then leads to the difference in soundscapes between reef and estuarine. So reef soundscapes are very loud and acoustically rich and diverse. Whereas the estuarine seagrass habitat that I used in my study was very quiet, it had to be amplified, and there were few biological sounds. There were a couple of snaps here and there, but that was it. This then consequently means that reef soundscapes and cues can be heard over hundreds of meters, whereas this soundscape was probably had a much smaller detection range. So the main takeaways from this are that this is the first study to examine estuarine larval behavior response to acoustic cues. There was an initial response to airgun noise in 3 of the 4 species, and then there was no preference or avoidance to the ambient. It'd be interesting to see in future studies if there is a difference in settlement size seagrass versus oyster larvae settlers, because an oyster soundscape is much louder than a seagrass as well, and more acoustically rich. And then it'd also be interesting to see if there's a difference in juvenile behavioral response as compared to these settlement size. And with that, I'd just like to thank all of the larval sources and the little larvae that were unknowingly used in this study. And I will take any questions.
Thank you, Emily. Great talk, and we have plenty of time for questions if anyone has a question. Terry?
Very interesting. Some of you who used to watch Laughing back in the day might know what that means. Anyway, I have a question regarding the source of your larvae, and maybe I missed it.
Sure.
Because I'm older and denser. Where did you get them?
I got them from TPWD, as
TBW.
Texas Parks and Wildlife Department. So they
Okay.
have a larval hatchery for
Okay.
the 3 species. And then I got the Florida Blenny from a lab at A&M Galveston.
Okay, the reason I ask is because some larvae don't recruit necessarily, but they land in places that are noisy thanks to humans to begin with. Others land in nice areas with salt creeks and what have you that only see motorboats and things like that. And so I'm wondering down the line if you plan to look at larvae that you can source from noisy environments versus, you know, like port areas or marina areas versus more pristine places, and see how those larvae react.
I mean, that would be very interesting. One other option would be to do like Simpson et al. Did and, you know, get them from TPWD, a quiet source, and then condition them and rear them in a specific sound and then see their noted behavior afterwards.
Yeah, you could do that too. So have fun with it. Yeah, thank you.
Thank you. Do you have a question, Joanna?
Yeah, you didn't actually, or maybe I was not paying close enough attention, mention the salinities that you were using and how salinities during rearing versus salinities used during the experiment might have contributed or not to basically sort of a lack of definitive action, it seems, on the part of these larvae. As somebody who doesn't really work in estuarine conditions, I would have thought that salinity would have really been a far more important trigger than sound, but that's just coming from somebody who, you know, lives and works in the middle of the Sargasso Sea where salinity doesn't change very much.
Yeah, that's a very good question. So in terms of these larval species, they tend to settle in seagrass beds. So I matched the rearing and the experimental tank salinity conditions to the salinity when I made my acoustic recordings, which was done during the time of settlement. So obviously, I guess I could have been affected had it rained, you know, previously the night before, or we hadn't gotten rain in a month. So that would be really interesting to look at. So I know that like fiddler crab megalobae, one of the species that settles like really far upstream, rely on a salinity cue as opposed to auditory or like chemical.
Thank you for coming to our second session of the day. It is entitled Emerging Issues, Climate Change, Marine Litter, Invasive Species, Stony Coral Tissue Loss Disease. So our first speaker this morning is Holden Harris, and he's going to be talking to us about the impacts of land use and climate change on coastal food webs and fisheries. Thank you, Holden.
Thank you, Kelly. Man, I'm excited to be back to GCFI in person, and I'm excited for the presentation today. My co-authors in this work are Dave Shigaris, Nate Reaver, Dylan Sinickason, Scott Alford, and Mike Allen. They're all at the University of Florida. I recently changed to the work for the University of Miami Cooperative Institute of Marine and Atmospheric Sciences and as a no affiliate, but this work was done at the University of Florida. I'm going to restart my timer. All right, we're live. So the theme of this talk is the natural resource management for what I'll call the land, ocean, aquatic continuum. So generally marine science and coastal managers focus on managing well marine and coastal resources. But if we zoom out from a biogeochemical perspective, the ocean is a sink that receives much of what we do on land. So it receives freshwater input, but also plastics and debris and toxins and fertilizers and carbon and nitrogen and phosphorus. All of this rolls downhill to the ocean. And to think about this, we can consider the big example in North America. There we go. The Mississippi River. The Mississippi drains 40% of the continental United States. It provides about 80% of the freshwater input to the Gulf of Mexico, as well as about 90% of its nutrients. And of course, this can be problematic when you have severe hypoxic events. In managing this terrestrial aquatic marine continuum, I think it's really, really hard. The system is about as complex as you can get. If you start to think about all the physical and hydrological and biological and ecological and chemical processes that are going on at the same time, as well as the socioeconomic components. Natural resource governance agencies are traditionally not well set up to maybe manage this either. So if you consider in the United States, a lot of upstream resources are managed by groups like the U.S. Department of Agriculture and the Department of the Interior and services like the U.S. Forest Service or the National Park Service, while downstream resources might be managed by NOAA and your state agencies. So what can we do in the face of this incredible scientific and management complexity? Well, if you're a modeler, you model it, right? And not all models are the same. Hope I woke you guys up. And of course, your model is going to be wrong, right? All models are wrong. Some models are useful. And our purpose here is not to capture the full extent of the complexity, but rather to reduce that complexity to something that's useful to us for managers. And so there's different approaches that we can go about this. I think an often undervalued one is the qualitative approach, with our objectives might be to better describe or illustrate and understand and communicate these complex systems. And then quantitative models also provide us, have much of the same objectives, but might allow us to do further things like state and validate assumptions to test hypothesis, and maybe try to forecast the future. So our objective here is to explore the impacts of climate and land use change on coastal ecosystems and natural resources. And we went about this in kind of two parts. So the first part is I'm going to present a conceptual model for this developed from case studies in the Gulf of Mexico. And the second part is I want to show you some work that we've started to link hydrologic and spatial temporal ecosystem simulations to develop these quantitative approaches. So to develop a qualitative framework for considering the downstream impacts of upstream natural resource management, we did 5 case studies in the northern Gulf of Mexico. So these include the Apalachicola Bay, Galveston Bay, Mississippi River, Mobile Bay, South Florida, and Swanee River. I won't go into all of these, but for instance, we already mentioned some of the issues involved with the size and extent and nutrients of the Mississippi River. Or I think a salient case study is the one of Apalachicola Bay. So in 2012, Apalachicola experienced a catastrophic collapse of its historic oyster fishery. And there's multiple stressors that are implicated. First of all, maybe overharvest or overfishing could be a possibility, but there's also documented hypersaline events, which increase predation and disease in this area. And the responsibility for this has been a major point of contention and has led to multiple Supreme Court cases with the state of Florida suing the state of Georgia. And I think these what are termed water wars kind of help show the potential management conflicts between downstream and upstream natural resources. Or we can consider the Swanee River estuary, which we'll talk about at the back half of our talk. This is a relatively undeveloped area of coastline, but is still experiencing considerable climate changes. We've documented increases in frequency and severity of droughts, as well as the system is tropicalizing. We now have established mangrove and snook communities in this area. So doing these, although each of those 5 watersheds are really unique, and have their own characteristics and challenges. I think it's possible, and it's also useful to try to tease out some of the commonalities between these. So the major driver for precipitation change is, of course, going to be climate, which is going to alter the quality and also the variability of precipitation. So in the Gulf of Mexico region, we are going to expect increased frequency and intensity of droughts, but also increased frequency and intensity of hurricanes. And beyond climate, we have local drivers. And so understanding these might allow direct management changes. The freshwater quality is going to be affected by things like pollution and nutrient runoff, and the quantity is going to be affected by land use and how we do management in that land use, considering municipal and agricultural withdrawals. So together, these climate and anthropogenic drivers affect the timing, the quantity, and the quality of water that's going to be entering our estuary, and these change the environmental conditions of the estuary. So salinity, turbidity, dissolved oxygen, temperature, that's all going to be changing. And of course, that's going to affect the ecology of the system. So that'll change your primary productivity, your community structure, and food web processes. And I think it's also going to affect our foundation species. And all this together can cause feedback loops. And all that together is real. The environmental health is going to be what's providing our coastal resources and ecosystem services. So the second part of our presentation, I'm going to give an overview of our current efforts underway to look at impacts of climate and land use change in the Suwannee River estuary. And unfortunately, I don't have final results to present to you. But I think for how I'm trying to design this talk, I think it can still be very useful to just mainly consider our approach to going about how we try to model this incredibly complex social ecological system. So there's three main pieces to our modeling effort. The first is our collaborators at the University of Florida Water Institute developed a hydrologic model of the Suwannee River Basin. Their inputs feed into an ecosystem model. That's what I've been working on. And together, both of our models are trying to help inform economic evaluations for cost-benefit analysis of management changes. So the first model piece is the hydrologic model. I mean, this is going to predict river discharge and nutrient outputs. And of course, the major driver for this, as we talked about, is going to be these ICP climate scenarios for precipitation. And for land use, we can consider current or changes in areas of cropland, of pasture, of forestry and urban development, and also how we conduct resource use in these areas and manage them. And these are going to result into changes in the quantity and quality of freshwater delivered to the estuary. So this is the Suwannee River estuary here, which is where our ecosystem model lives. The approach to our ecosystem model was an ecopath with ecosystem framework. So ecopath is a static mass balanced model. So basically I build a food web of all the critters and things in this system and what's eating what. And then in EcoSim, we simulate that over time and fit these to large data sets. In EcoSpace, we can make this spatially explicit. And this is what we're doing the future projections in. So the figure here shows the Suwannee River estuary food web in our ecopath model. So these big brown things on the right, these are detrital groups. On the left, you see our primary producers like macroalgae and phytoplankton and seagrasses. And these connections, as you see, is the bioenergetic links of how energy moves up from the producers to primary and secondary and tertiary consumers up to important fisheries groups and eventually into our 8 fishing fleets, which informs the economic evaluations. So in EcoSim, the food web is fit to long-term time series. And quickly, I'm going to show you some plots. Don't worry about trying to interpret these. I'm not going to orient you to them. Or rather, I just want to give you an idea of some of the extensive data that went into fitting our model. So probably the most important data set is information collected by decades of sampling by FWC's Fisheries Independent Monitoring Program. And this helped estimate our biomass densities. I use this data also to develop environmental preferences for the species in the model, which helps spatially orient them. Catch data was provided by a NOAA MREP program for fisheries recreational monitoring, as well as fishing effort. River flow is monitored by the U.S. Geological Survey. Nutrients has been monitored in the area by the University of Florida, as well as oyster reefs by University of Florida's Lone Cabbage Reef Restoration Project. In aquaculture information, we gathered from the Florida Department of Agriculture and Consumer Services for the region's important shellfish aquaculture industry. So you can see this is a big multi-agent effort, and we take all this data and it all goes in to these models. And so this is what it looks like all together. It's pretty messy because it shows all 6 functional groups. But these are changes in the relative biomass density compared to our reference year of 1997 and then going over time. What you can see in these pink things that are going up, these are snook that are establishing in the population as kind of an invasive species. So relative to their initial biomass, they're increasing a lot. And on the bottom is zoomed in. You see the lower trophic groups in red going up and down, which is driven by nutrient inputs from the river. And we can zoom into these and do a lot more analysis with this. So this is just showing some of the important recreational fisheries in the plot in our model outputs of trying to fit the observed data, which is those dots there. So up on the top, we see that the model does a pretty good job of capturing like this invasion of snook as snook move north established in the region. And it also captures the concurrent declines that we've seen in other recreational fishery groups like red drum and spotted sea trout. And so I'm disappointed that we don't have more results ready to show you from this ecosystem model, but I still think there's really some important lessons that emerge from the approach. So first of all, it's apparent that the ecology of the system is driven by changes in the quantity and quality and timing of water delivered. And so management is going to require holistic stewardship of these watersheds and wetlands. And second is that we can really expect compounding impacts from climate change. So this is something that Bob Glazer talked about at the start of our conference in his keynote address. Climate is affecting every part of the system, and the effects can be multiplicative, so they can multiply against each other. So if we consider drought, for example, drought is going to cause lower freshwater discharge into the system, but it might also increase municipal and agricultural use of water together. It's also a hotter climate is also going to drive more frequent and severe heat waves and also continue to redistribute species. And these environmental changes are going to be observed on our foundation level. So things like our corals and marshes, seagrasses, and oysters. And historically, what we've seen is that the loss of these foundation species can be tipping points that can change the ecosystem to an alternative and often degraded stable state. So altogether, understanding and forecasting this land-ocean aquatic continuum crosses many scientific disciplines. And I think this is an exciting area, a frontier of research, because we're able to model the system in novel ways that, because of advances in technological and computing capacity, that we really weren't able to do so up until now. That said, we really need to recognize that these big and increasingly complex models also increase our uncertainty with them. And the lesson to me is that we have to be very careful and thoughtful for how we go about designing these models and really think through our objectives and scope. And I think to help with that, it's absolutely important that you get stakeholder engagement early and often, and that co-development, I think, is really going to be needed to make sure these models or results and the data products provide something that's valuable. So thank you very much. I'm really happy to be back. I want to mention that our research is supported by a grant from the National Academies of Sciences and also from the US CAST, the Climate Adaptation Science Center. There's my contact information. So thank you very much.
Thank you, Holden. Unfortunately, we don't have time for questions, so you guys can check Holden at lunch. Take one. You need a minute. All right, we can take one question because Fadila needs a minute. Yeah, go ahead, Bob.
Thanks, Holden. That was really interesting. I have a question. It's not going to have anything to do with the science. But at the beginning, you mentioned the ineffective governance structures that probably are impeding the ability to actually have effective management. And I'm wondering if you could speculate on that, because that, in our experience, that's always the barrier that you have to overcome is governance based. And you know, we could produce the best science, but if the governance structures aren't there, then we're not going to be able to accomplish any meaningful management approaches.
Yeah, it's something that I've thought about, and unfortunately, I'm not sure how much insight I could give, because it's something that I've worried about this from the beginning of this project is what are the management outcomes from this? Who is going to take this and how are they going to, what are they going to do with it? Because it's not really clear. And something that someone told me that's, you know, is more experienced and works with this and says that I think has been working for a while and says sometimes science does go ahead of management. And so maybe you don't necessarily need a clear objective, management objective out of this. And so that made me feel a little better, but we'll see how it all goes.
All right. Thank you so much, Holden. I realized earlier I didn't introduce myself. My name is Kelly Kingan. I'm from the University of Trinidad and Tobago. Our next few talks are going to be virtual. So the first one is by Sebastian Herrera, and it is on blue carbon stalks and mangroves of an oceanic island biosphere reserve in the Caribbean Sea.
Hi, welcome to my presentation. I am Sebastián Herrera Fajardo, and I'm going to speak about blue carbon stocks in mangrove of an oceanic island biosphere reserve in the Caribbean Sea. Below, you can see the people that work with me in this project. We are from the Instituto de Investigaciones Marinas y Costeras José Benito Vives de Andrés in Bemar from Santa Marta, Colón. The mangrove blue carbon is one of the most important stories nowadays. It refers to the capacity that the mangrove forest has to capture and store the organic carbon in this ecosystem for a long period of time, which is taken by the trees as dioxide carbon, and then is stored in the underground biomass and above ground biomass, necromass, and the soil components. In this way, the mangrove ecosystem contributes to mitigate the climate change, and it allows to make strategies to conserve the mangrove because of the land use of change, and of course, they have an important aspect related in the ecosystem services as a barrier against hurricanes, coastal erosion, and improve the high diversity. Also, the blue carbon mangrove could allow to prove available source of future economic growth through the carbon trading by the sequestration and storage services. Besides, the lack of data in the carbon quantification on islands, and also the marine protected area are key tools to conserve biodiversity and mitigate degradation coastal ecosystems like mangrove, and of course its stored carbon capacity. For that reason, our goal was to determine the blue carbon in a marine protected area in the forest mangrove of San Andrés Island. This is San Andrés Island. We make our investigation in the north part from Colombia. In the right figure, you can see the biggest island is San Andrés in the right part of the figure. This is located in the Sea Flower Biosphere Reserve that was an archipelago, and also it was inside the marine protected area. The mangrove land cover in San Andrés is 134.9 hectares, and San Andrés was an akarstic island in the Caribbean Sea. It has an 27 km square, and we have found an average annual precipitation of 1,173 millimeters. So we can find in this island the mangrove mainly in the east part, the located the mangrove. So we can find different types of physiographic mangroves. We have found basin, French, and inside forest mangrove. We use 21 circular plots from an area of 452 meters squares for calculate the carbon stored in this ecosystem. We measure above ground biomass for calculate the forest structure by the DAP and the height trees. We use analometric equations for calculate the biomass in this forest. We use Medina's works. We calculate the necromass by fallen wood and trees dead standing, as Kaufman et al. Recommend. And we calculate the soil in the soil carbon storage. We take a core with a Russian pet border from 0 to 30 centimeters for calculate the organic carbon concentration and soil bulk density that was calculated in the LabCam laboratory from Invertebrates. Then our results in this project, we found that the total carbon stored in this mangrove was 300, more or less 47 megagrams of carbon per hectare. The highest carbon store was from the soil that was represents the 60% of organic carbon of the organic carbon in this island. The second component was the above ground biomass with 122, more or less 25 megagrams per hectare. And the last was the necromass with 9, more or less 2 megagrams carbon per hectare. These results contribute to prevent the land use and the land change because of in this island have a loss cover for human pressure like constructions, livestock, and discharge water, was water. And also the carbon stored in this island is less than the Colombian continental mangrove and mondial average, but it is important to take in mind that are from an island that doesn't have river or spawns. We found in this story that the first report of carbon stored in San Andres mangrove in the above ground biomass, necromass, and soil until 30 centimeters deep, and these are comparable with the reports of continental mangrove and the world average, taking into account that the previous ones correspond to a depth of 1 meter in the other studies. The total organic carbon store of the mangrove of San Andres was lower than the world average, and in general lower than the record in the Colombian Caribbean mangroves and greater than the Pacific ones. However, it is important to highlight that the measures made in the present study correspond to the first 30 centimeters of soil depth, in contrast to the other records that were obtained at greater depths, which demonstrate the importance of the mangrove present in these oceanic islands that have conditions in the water regimen different from the continental mangroves. This is the acknowledge to Coralina that was the support for our project, and the Ministerio de Medio Ambiente y Desarrollo Sostenible from Colombia, of course, Invertebrates, and this is the team that works in this project in the field trip. Thank you for your attention.
Thank you so much, Sebastian, for the information on carbon storage and mangroves in San Andres. We have plenty of time for questions. If anybody has a question for Sebastian, he's online. Yeah, Marta?
Hi, yes, I am here. Can you hear me?
Yes, we're hearing you. Marta's going to go ahead with her question.
Yes, thank you, Sebastian, for your talk. It is nice to see how these kind of studies are being now conducted in the archipelago. So my question for you is, considering that the mangroves coverage in San Andres Island is relatively small, and they have been suffering from degradation for many years. These low values that you are describing today are in a way related with that pattern of degradation, or that consider these mangroves as a forest in recovery? Thank you.
I think that the degradation of the cover of mangrove ecosystem in San Andres Island was not the reason that we have less carbon store in the island, because of this is a small land compared with the continental zones. So in this case, the reason is more of the quantity of an area of cover that the mangrove have in this island. But it is important to take in mind that these forests, these mangrove forests, have the capacity to grow without hydrological regimen permanent. Okay, so it's important because of this, different from the continental mangrove that have rivers or another water bodies that exchange the salinity to low this physicochemical abiotic parameter.
Okay, thank you, Sebastian. We have some more time if there's any other questions. Anything virtual? No? All right. Well, I guess if that's it, we will move on to the next virtual presentation, which is coming from Trinidad and Tobago. And this is Jesse Williams, who will be assessing vulnerabilities to climate change using a rapid vulnerability assessment.
My name is Jesse Williams, and I will be presenting on the topic, assessing vulnerabilities to climate change in Trinidad and Tobago using a rapid vulnerability assessment. I will be presenting on behalf of myself and Dr. Sharon Hutchinson from the University of the West Indies, St. Augustine, Trinidad and Tobago. Here are some of the table of contents that will be covered in this presentation. Begin on the global agenda, climate change is at the forefront of most discussions at the multilateral level. The effects of climate change are systemic in that it doesn't only impact natural resources, but it impacts those who depend on such natural resources. Our Ministry of Agriculture and Local Fisheries issued a press release in 2017 where it is stated that the fisheries and marine industry accounts for employments both directly and indirectly through sales, repairs, preservation, marketing, etc. In light of the above, monitoring climate change has to be of utmost importance. The article further goes on to state that the very livelihood of coastal communities are threatened and they will bear the major fallout if systems are not put in place to reverse climate change. The FAO, in its publication, Addressing the Climate Change and Poverty Nexus, sets the approach in achieving such. Mainstreaming climate change in pro-poor or social policies is heavily encouraged, and vulnerability assessments are the building blocks to addressing this nexus. It is important that we look at this nexus because in Trinidad and Tobago, research done by the ministry shows that there is overfishing in our waters. And of course, by reading the literature, we know that climate change could exacerbate existing vulnerabilities that exist in communities. So in the literature review, some key points stood out in that, one, climate change and its impacts on the socio-economic landscape can be complex. There's urgent need to develop mitigation and adaptation strategies suited to the local context. And in my literature review, I came across a framework done by FAO and Canary, which is a toolkit for vulnerability and capacity assessments. And in this toolkit, there's provision for a rapid vulnerability assessment. And the tools for this assessment include the likes of hazard trend analysis, seasonal calendar, problem tree, rapid community mapping, etc. Climate change is modifying the distribution and production of marine and freshwater species. The consequences of sustainability of aquatic ecosystems for fisheries and aquaculture and for the people that depend on them are uncertain. The very livelihood of coastal communities are threatened, and they will bear the major fallout if systems are not put in place to reverse climate change. Hence, we've looked at a community in Trinidad and Tobago bordering the Gulf of Paria, and that community is Orange Valley. One, it is a coastal community. Two, it is a community dependent on fishing as their livelihood, and by the fact itself, having these characteristics makes such a community vulnerable to increasing incidence of climate change. So some of the research questions are, what is the vulnerability context of residents in Orange Valley? What systems or strategies do households have in place to deal with the effects of climate change? And what are the factors that affect vulnerability? So here's a methodology that was used to undertake the research. We did a rapid vulnerability assessment, and this was preceded by a desk review into Orange Valley, looking at the vulnerability context of Orange Valley under the Sustainable Livelihood Framework, which was one of the frameworks identified in the toolkit developed by FAO and Canary as a framework of analysis when doing the rapid vulnerability assessment. On the ground itself, that is the rapid vulnerability assessment, a hazard trend analysis was conducted through a workshop and semi-structured interviews through focus groups. The sample was purposefully selected to ensure that a broad spectrum of views were considered in the research. For example, we had a focus group specifically for women alone, and we catered for young adults. The research was conducted in collaboration with the Orange Valley Village Council. And here are some pictures of community engagement. The preliminary results are that the community is seeing potential fish migration. Well, it's seeing fish migration as fishermen and wives of fishermen are complaining that it is getting harder at sea and that there's less catch in the grounds that they normally occupy, meaning that they have to go further out to get a good catch. There have been storm surges in the community in recent times, most recent as 2018, where a storm surge affected one of the cultural assets in the community. And there have been more flooding in areas that were not prone to flooding before. And what came out in the engagements workshop with the community and in the focus groups were some interesting local adaptation strategies to combat the effects or the impacts of climate change. There has been reclamation of land in the community, and the household and most households always keep a stock of fish frozen in periods in case when there are periods that there's low catch, they always have a food source for sustenance and for sale if the case may be. What was clear is that there exists poverty in the community, and I would say relative poverty and instances of transient poverty due to the variable nature of fishing currently. So some issues that exist were high school delinquency, access to food food prices, cancer, and of course, income from fisheries, where incomes have been very variable in recent times. To conclude, definitely there needs to be intervention in the community, and such intervention could be training with respect to value addition so that fishermen perhaps can extend the shelf life of food and reduce or mitigate their instances of transient poverty, and by the fact itself and by extension, reduce the effects of climate change. Thank you.
All right, thank you, Jesse, for informing everyone of flooding and catch issues with the fishing community in Trinidad. We have a few minutes for questions, if anybody has any questions for Jesse. Going once, twice, 3 times. All right, well, I don't see any questions, so we will move on to our next
Buenos días. El trabajo.
Sorry. Our next virtual talk, which is by Dalila Aldana Aranda, and she is going to talk to us about microplastic pollution and green turtles in the Caribbean.
El trabajo que voy a presentar lleva por título.
For those of you that weren't here, the tutorial and interpretation, you can use the app. So you'd have to join, go on the agenda item on the app, and you'd have to use your own personal headset. And to the bottom right, there'll be 3 dots. If you click on that, you'll see the interpretation function, and then you can choose English. So I'll give you all a couple seconds and then I'll press play.
Por microplásticos en la tortuga quelonia Midas del Caribe mexicano, que es un trabajo en equipo con los autores aquí indicados en esta primera imagen. Como sabemos, la producción de plástico aumenta y en los últimos 20 años ha producido más de la mitad de todo el plástico que hemos producido, por lo que anualmente llegan a los océanos una descarga de 9 billones de toneladas de basura, de los cuales el 80% son plásticos. Y de estos, más de la mitad son plásticos de un solo uso o de embalaje. Un ejemplo, países como Trinidad y Tobago, Antigua, Barbados, Santa Lucía, Bahamas, Granada, generan una cantidad de plástico superior al peso de 20,000 transbordadores espaciales, es decir, 40 millones de toneladas de basura plástica en estas islas. En relación a los microplásticos, sabemos que estos provienen principalmente del lavado de nuestra ropa, que mucha de ella es sintética, y del desgaste de nuestras llantas, de los autos principalmente. Por otro lado, tenemos que los microplásticos pues están omnipresentes, que tienen un efecto tóxico, que están relacionados a varios tipos de cánceres, que son vectores de otros patógenos, que concentran y acumulan metales pesados y otros compuestos químicos organoclorados, y que esto tiene consecuencias en la gestación de otras especies marinas y del propio hombre. Tenemos además que el Caribe pues es una zona que que representa 35 países, 3 millones de kilómetros cuadrados. De aquí proviene el 11% de la captura de pesca mundial y es un destino turístico internacional esta zona. Por otra parte, tenemos que el Caribe es uno de los mares más contaminados del planeta Tierra, de acuerdo a Naciones Unidas, y esto debido a 3 factores. El primero es la no disposición de la basura en sitios oficiales, que el 85% de las aguas residuales no son tratadas y que prácticamente no hay capacidad de reciclado en la zona. Con estos antecedentes nos planteamos el objetivo de conocer el grado de contaminación por microplásticos en esta especie de tortuga. Como sabemos, esta tortuga en los estadios de neonatos y juveniles tienen una alimentación omnívora, pero en la fase adulta esta especie es herbívora, principalmente de algas y de pastos marinos. Y tenemos que en los pastos marinos ya tenemos reportes de la presencia de microplásticos en ellos. Por otra parte, tenemos que esta tortuga es una especie como las otras tortugas amenazada, está protegida de manera internacional y en cada uno de los países, por lo que no se puede sacrificar ninguna de estas especies. Con base en esto, nosotros trabajamos con un método no invasivo, utilizando en este caso las excretas de las tortugas. Estos heces de las tortugas se colectaron en el norte de la península de Yucatán, en Isla Blanca y en la laguna arrecifal que se encuentra en esta zona. Las heces de estas tortugas se colectaron tanto en mar como en la playa. Cada una de estas heces colectadas se tomaron 3 subréplicas, cada una de estas de 1 gramo, y estas muestras de heces de tortugas se sometieron a 2 niveles de digestión con agua oxigenada al 30%, y en cada una de estas, de las soluciones obtenidas, se llevó a cabo el conteo y la clasificación de microplásticos con microscopía óptica. Para determinar el tamaño de estas microfibras de plástico, se utilizó microscopía óptica y el programa Image Pro Plus. Para los análisis químicos, la morfoestructura de las microfibras de plástico y el tipo de polímeros, se asoció la microscopía electrónica, el análisis EDX, la microscopía Raman y la microscopía y análisis IRTF. Como resultados, encontramos que todas las heces analizadas de esta especie de tortuga, tanto las que provienen del mar en azul como las que provienen de la playa en amarillo, todas presentaron contaminación por microplásticos. En términos generales, la concentración de microplásticos en las heces colectadas en el mar son 5 veces superiores a las encontradas en la playa, y esto por procesos de erosión expuestas al aire. Por lo que respecta al tamaño de estas fibras, aquí tenemos una clasificación en rangos de 0 a 1000, de 1000 a 2000, etcétera, hasta 4000 a 5000 que corresponden a la clasificación de micropartículas de plástico. Tanto las fibras provenientes de tortugas en el mar como las tortugas, las heces de tortugas de la playa, la mayor parte de estas fibras están en el rango de tallas de las 2000 a las 3000 micras. Por lo que respecta a los colores de estas fibras, el 100% de los microplásticos encontradas en las heces de tortugas son fibras. Y el 75-80% de ellas corresponden a fibras azules y fibras transparentes. En este gráfico tenemos los análisis químicos de estas fibras. Aquí tenemos fibras azules, transparentes, rojas y negras. Y para cada uno de este conjunto de fibras de esos colores se llevó a cabo análisis químicos EDX. ¿Qué encontramos? Encontramos que en todas ellas, independientemente del color de la microfibra, está presente el cloro y el azufre. Esos dos elementos son indicadores de absorción de fertilizantes. Por otra parte, en todos ellos encontramos también metales pesados. Esto está asociado con las pinturas que se utilizan para colorear los plásticos, con excepción, obviamente, de las que son transparentes. Lo mismo hicimos por rangos de tallas para diferentes colores y encontramos que todas presentan cloro y azufre y hubo una concentración más abundante de fierro en las fibras que son de mayor longitud. En este gráfico tenemos dos ejemplos de la ultraestructura de fibras y el análisis químico con EDX que nos permite ver la presencia en un caso de cloro y en otro caso de flúor, y estos dos elementos se asocian a polímeros que pueden ser potencialmente PVC y PVF. De una manera más precisa, con el uso de análisis IRTF y Raman, aquí tenemos dos ejemplos, una fibra azul y una partícula negra de estos microplásticos. Los espectros que están en la parte inferior corresponden en ambos casos, tanto para la fibra azul como para la fibra negra, a el polímero nylon. Este nylon proviene muy posiblemente de la ropa que utilizamos. En este caso tenemos una imagen en la cual vemos un detalle de una microfibra con la presencia de una diatomea, en este caso un detalle mayor de el desgaste de una microfibra, y dentro de este ya vemos la colonización por otro tipo de microorganismos. Observen la escala, aquí estamos hablando de un microorganismo de 10 micras. Aquí tenemos otra fibra con detalle, colonizada ya esta fibra por partículas calcáreas, partículas de sílice y la presencia de diatomeas que comienzan a colonizar esta fibra. Como conclusiones, todas las muestras obtenidas presentaron contaminación por microplásticos. En este estudio, la concentración de microplásticos es muy superior a la reportada por otros autores. Por otra parte, todos los autores que han trabajado en tortugas lo han hecho principalmente con tortugas a partir de necropsias de animales que llegan muertos a la costa, y a partir de ahí trabajan, con excepción de un autor que también ha trabajado con las heces y nosotros. En todos los casos, las micropartículas de plástico que encuentran son fibras en todos estos casos, y esto asociado pues a nuestros textiles y las aguas residuales de descarga. Entonces, el lavado de ropa, las aguas residuales no tratadas llegan a la laguna arrecifal, se sedimentan y se adhieren, entre otros, a los pastos marinos, y pues en esta zona es donde se alimentan estas tortugas en la fase adulta. También hay que pensar en el tipo de corrientes regionales a nivel del Caribe y las locales a nivel de las lagunas costeras, y por supuesto también la presencia de embarcaciones, dado que esta es una zona importante de navegación turística y pesquera. Y el nylon, pues ustedes saben que también está asociado a las artes de pesca. Por último, estos estudios de microplástico, vemos que con los métodos no invasivos que comenzamos a poner en detalle con nuestra especie modelo del Caribe, que es el caracol rosa, pues son útiles para otras especies amenazadas, como es el caso de las tortugas. Y recordar que para volver a vivir así en el Caribe, para volver a tener estas playas, pues tenemos que pararle a la producción de estos plásticos y por supuesto a su consumo con acciones responsables y sustentables. Muchas gracias por su atención. Tienen en pantalla mi nombre y mi correo electrónico. Gracias.
All right, thank you so much, Dalila, for a very comprehensive talk on sea turtles and plastics. I think we have a couple minutes for questions, if anybody has.
Buenos días. El trabajo que voy a presentar lleva por título Contaminación por microplásticos.
Oh, we got a virtual question, all right?
Dalila, Claire Morale from Grenada is asking you, did you find any cotton fibers in the turtle samples?
Okay. Okay. Thank you. Thank you for the question. Please, it is possible to repeat the question. I heard the first part, but not the second section of this question.
She's asking, did you find any cotton fibers in the turtle samples?
The fibers?
Cotton fibers in the turtles.
No, no. I will explain my answer. When you prepare the sample, you use the chemical substance that dissolve the organic components. So it's possible to find the cotton fibers, but the cotton is made with the acid that we use in this process. So the only fibers that we found in these turtle samples are, in this case, nylon.
Okay, thank you. Any other quick questions for Dalila? No? Okay, well, thank you so much. Gracias. Our next talk is by a colleague of mine in Trinidad, Ladena Kanhai, and she is going to talk about the human dimension of plastic pollution in Trinidad and Tobago.
Hello there, everyone. My name is Ladena Kanhai, and I'm a marine scientist that is based here at the Department of Life Sciences at the University of the West Indies at its St. Augustine campus. And I am really delighted to have the next couple of minutes to share with you some of the research that I am currently involved in as it relates to the human dimension of plastic pollution in Trinidad and Tobago. I do want to take this opportunity, of course, to acknowledge my collaborators on this project. So we have Ellis Keller from the UK, and we also have Isabel Richter from Norway. So what we do know as it relates to plastic pollution in the Caribbean large marine ecosystem is that we have a problem. We have got 4 decades of data that tell us that we have a problem. To some extent, we also do know that the plastic debris that is present in the Caribbean region is having a negative impact on biodiversity. And of course, we understand, for the most part, what is causing the problem. But I think that if we are to develop interventions in this UN Decade of Ocean Science, that those interventions must be based on sound science, okay? Many of you would, of course, be quite familiar with the fact that Trinidad and Tobago are the most southerly islands in the Caribbean archipelago. What we know here in Trinidad and Tobago is we have a problem with plastics. Since 1994 to now, volunteers have participated in these citizen science-led ICC cleanups. And what they have reported is that plastic debris is accumulating in our coastal and marine ecosystems. Okay, we also have some snapshot studies that have just indicated to us that the plastic debris is fragmenting into smaller particles, and so now we have microplastics that are present in the environment. Here in Trinidad and Tobago, we do have interventions at the level of the government, at the level of non-governmental organizations, private sector, individuals. But even though those interventions exist, we still have a problem. And that is really what provided the rationale for our study. So we wanted to delve a little bit deeper into the human dimension of this issue. Specifically, we wanted to assess the behaviors of Trinbagonians as it relates to their use and their disposal of plastics. And also we wanted to understand what was really driving behavior. All of this in an attempt to, at the end of the day, be able to develop a catalog of systematic psychology-informed interventions that could be, tailored to specific behaviors, and then, of course, to specific stakeholder groups. So to do this, what we then did was that we started off by designing a questionnaire. So myself, with my expertise in the area of plastic pollution, and then Isabel Richter with her expertise in environmental psychology, we sat down, we designed a survey. We, however, acknowledged that there were many local experts that were working in various sectors that could validate this survey for us. So we sent out the survey to them, requested that they validate it, help us to improve the content, help us to improve the clarity of the questions, right? They indeed provided us with really valuable feedback, and we used this to be able to revise the survey. Of course, the questionnaire was screened by the Campus Research and Ethics Committee, and we did have approval to commence the data collection. So once the survey was open to the public, we employed multiple channels to get, to publicize that this survey was open. So we tried the social media platforms. We, of course, targeted organizations that had these well-established distribution networks, and the survey was open for 15 weeks. Right? We're still in the data analysis phase, but what I am presenting to you would be some of the preliminary findings. Okay, so in terms of the socio-demographics, what we noted was that generally we had about 533 participants. Okay, the majority of them would have lived, been residents of Trinidad, so about 97%. The majority of them would have lived in the country for their entire lives. And the majority as well would have identified as females. Okay, what you would notice is that generally the distribution amongst the different ages would have been well distributed. And of course, you're seeing here that the majority of participants would have had tertiary level education. Now, the participants of our survey, they indicated that they did regularly spend time in nature, and that when they were there, they often noticed litter. All right. So in terms of plastic usage and recycling, many of the participants indicated to us that the items that they intentionally bought and received with purchases on a regular basis would have been things like the plastic bags, the plastic beverage bottles, the COVID-19 protective equipment, and even the plastic takeout and takeaway containers. Those that they intentionally bought and were, received with purchases on a less regular basis would have been plastic cutlery, plastic straws and stirrers, the plastic cups, and the plates, right? We also wanted to find out why people use plastics, all right? And so what we're seeing here is that people are generally using plastics because they are convenient, because they do not cost a lot of money. We also see that people are using plastics out of necessity. So this might be connected to hygiene, this might be connected to their need for access to purified water, right? That sort of thing. But we also note that some people mention that they felt as though there was a lack of other options. And that is why they were using the plastics, okay? So we also focus on the issue of recycling in Trinidad and Tobago. And we wanted to find out from people, were they familiar with the current national recycling programs? And what we are seeing here is that 79% of our participants indicated that yes, they were familiar with these programs. But out of these, only 50% indicated that they were actually using these programs through their workplace or the community, right? It is important to recognize that approximately 37% of the participants indicated that even though they weren't using the program, that they would be very much interested in using or recycling, right? When we asked about what were some of the barriers to recycling in Trinidad and Tobago, we noted that there would have been systemic barriers. So people felt that, you know, there's no curbside collection of recyclables in my community, or that the collection bins, they're too far away. What is important to note is that knowledge was not a strong perceived barrier to recycling, right? And so the point I do want to make here is that if we keep pumping resources into public education and awareness and do not simultaneously pump resources into improving infrastructure, dealing with the system, we possibly might keep on seeing these trends, okay? We also wanted to find out from people a little bit about their perception of environmental damage. So we asked people, you know, what did you think was the biggest negative impact on Trinidad and Tobago's environment? And you would see that they pointed out things like the dumping of garbage, plastic pollution, industrial pollution, quarrying. It's important to know that even though they identify things like climate change and, you know, the development of infrastructure and housing, climate change didn't actually rank in those top three activities. Yeah, there were also activities that they did identify, things like agriculture, tourism, and fishing, of course. Yeah. In terms of for people, you know, what were the biggest or what were the drivers really of plastics in the marine environment? What we did see was that people identified issues such as the lack of law enforcement, the intentional dumping of waste, the popularity of single-use plastics, you know, the intentional littering, right? People also identified a lot of sort of systemic issues, a lot of infrastructural issues. And then they would have also, at the other end, they would have also identified some unintentional drivers of plastics in the marine environment. So things like wind and water distributing plastic litter, the loss of fishing gear in the ocean, right? That sort of thing. We also asked people to indicate, you know, to what extent you thought the following factors were, of course, affected by marine plastic pollution in Trinidad and Tobago. And to no surprise, people identified marine biodiversity, right? Recreation, cultural activities, aesthetics, fishing and seafood supply. What is important to point out here is how low human health was ranked, right? So that's something to pay attention to. We also in this project wanted to investigate behavioral intentions of the participants. So we asked them, you know, how likely would it be that they would support the following policy interventions? So a deposit refund system, for instance, for returning plastic bottles, people indicated that they were likely to support something like that. A ban on single-use plastics, they were likely to support that. Even for things like a monthly fee for the curbside collection of recyclables and even a tax on single-use plastics, people identified that they were likely to support policy interventions like that. Okay. We also asked people, you know, to think about maybe in the next 12 months, how likely were they to engage in activities, right? And what we saw would have been that people were very much interested in changing their consumption patterns to reduce the use of plastics. People who also, you know, they indicated that they would support, you know, environmental organizations that are actually already tackling this problem. Many people indicated that they, you know, of course, would be willing to influence people within their sphere of influence, right? And then, of course, what we saw was that participants were less willing to potentially participate in beach cleanups or to, you know, pick up litter on the street. So essentially to remove plastic waste from the environment, okay? And then finally, we also did want to have a look at, you know, what was actually driving the behavior of people as it relates to plastics. So was it knowledge? Was it social norms? Was it habits? You know, level of concern about the issue, right? The sense of responsibility that some people felt. And I will say that we are in the process of developing models. We do want to ensure that we are interpreting these models properly. So we will not share them at this point, but it will be something that we will release at some point in the future. And so why are studies like this important? From my perspective, studies like this are important because we need, you know, when we're developing interventions, we need to delve a little bit deeper and look at the human dimensions of some of these problems that we are developing interventions for. And I think that many of the points that were discussed in our presentation, they can be used to tailor some of the interventions that we can potentially implement on a national scale. So I'll leave it there for now. I'm happy to take questions in the, you know, after the presentation. And so I'll end the recording here. Thanks very much for your time.
Thank you so much, LaDana. It does not look like we have time for questions for her, but you should be able to contact her. I don't know if you want to. Yeah. Oh, you want to take one? All right. I see one question. Been told I can take it, so go ahead.
I enjoyed the presentation. Your questionnaire, as well as the results, sort of buy in, or at least seem to be based around sort of the narrative that it's individual consumers and local governments that bear primary responsibility for plastic pollution, which is a narrative that the industry has done a great job in perpetuating for nearly 5 decades. Did you ask any questions at all that looked at issues such as the you know, corporate responsibility for plastic pollution and looking at policy actions that would require more remediation on the part of companies? Or was your focus entirely on the role of individuals and local governments?
Yes, thanks very much for that question. First of all, can you all hear me?
Yes.
Yes. Okay. Thanks, Kelly. Yes, thanks very much. So I acknowledge that point that you made. You know, when we thinking about this issue of plastic pollution, very often we tend to think about the, what's potential solutions. You know, what are these solutions? Does the burden fall on the consumer? This particular questionnaire was sent out to Trinidad and Tobago. So it was not tailored to a specific stakeholder group. And it is for that reason, we didn't ask questions that would have targeted companies. You know, we could not tell who would take our survey. As you saw, you know, we sent this out to any resident of Trinidad and Tobago, 18 years and over. We only got 553 responses. So we did not specifically include questions that tackled interventions at the level of companies. I think what could be done, however, in the future is that a specific survey could be tailored to that particular group. And then when, of course, we're dealing with the issue of plastic pollution, of course, we're thinking also of upstream solutions. So even not at the level of, okay, so we have a problem, how do we deal with that problem? But we also need to examine even before the product is sort of designed, you know, and that's a whole different ballgame. But no, to answer your question, we didn't ask any questions, that tackled companies specifically and their responsibility as it relates to the issue.
Excellent. Thank you so much, LaDana. Great question. And I just wanted to say that some of the changes in Trinidad actually have come from the corporate level down and have made a really big impact. So even though this was directed at that, I think some of that is happening on the positive side. So, all right. So we will move on to our next presenter, who is also from Trinidad. And you guys probably all know her. Fadila Ali is going to talk to you guys about introducing the Global Partnership on Marine Litter digital platform. Sure. Yeah.
Hello, everyone, and good morning. So now I have to deal with this. So my name is Fadila Ali. I am from, I'm the Assistant Executive Director of GCFI and also the Program Manager for the Global Partnership on Marine Litter at the Caribbean Node. So a little bit about a background about the Global Partnership on Marine Litter. It is a partnership, and wow, I cannot see because I'm blind. It is a multi-stakeholder partnership consisting of government, NGOs, civil society, youth, essentially everyone working together to reduce or prevent marine litter and reduce the leakage of marine litter, and also focusing on the 3 R's, which are reduce, reuse, recycle, but also encouraging the steps before that. So to refuse, to repurpose, to upcycle and those sorts of things before we even get to the reduce, reuse, and recycle part. And it also is to support the implementation of relevant meetings such as UN General Assembly or UN Environment Assembly, as well as the 2030 agenda. And a little bit about the Caribbean Node. The Caribbean Node is co-hosted by GCFI, as well as UNEP's Caribbean Environment Program. So we work directly with UNSAP to implement lots of activities. We have lots of partners related to academia and youth. We've worked with governments, and we're all, we've supported projects within the Caribbean, as far as Brazil as well, because we are focused on the wider Caribbean region, and Brazil has a lot of influence on marine litter activities. So some of the aims of the Global Partnership on Marine Litter digital platform, which is what I'm presenting on today, is firstly to identify any gaps and priority actions for marine litter prevention and reduction, and also to integrate data and information from multiple sources. It is sort of crowdsourced and open source data that we're looking for. And also to coordinate and guide action based on the best available knowledge. So also there's a lot of work for action plan development and also to connect stakeholders. And as well with facilitating target setting and measuring progress. So in terms of the multi-stakeholder engagement, oh, I really can't see. There are lots of sponsors and lots of stakeholders involved, and the bit on the right are logos of all the different partners that are involved, as well as the different stakeholders are on the left. And then on the bottom right are the different sort of priorities of the digital platform, which are science policy, national action plans, guidelines, standards, and harmonization, sustainable and innovative financing, and making it accessible to all. And in terms of the progress of the digital platform, it is in multiple stages right now. So far we're approaching phase 5. Wow. So for context, I am legally blind. My prescription is like -8.5, so I really cannot see. And I can't see up here either. Yeah, so we're entering the phase of driving change by innovation, and that will be in February 2023. And I hope you all can see this because I can't. And this is about the membership of the GPML, and this is really just to highlight. So the GPML is a global effort, and so within the Caribbean, Latin America, and the Caribbean, Gulf of Mexico, whatever, we would be sort of under LAC. And as you can see in the bottom left, no, top right, there are only 7 governments that have signed up for the Global Partnership on Marine Litter. And in terms of the different types of organizations that are involved, you can see that it's mostly civil society, and there's a big lack of, there are less of the governments involved. And in terms of the regional distribution, I think that's 55. Could be wrong. There are only 55 entities from the LAC. And so far, in terms of what has been achieved, there have been 2,000 resources. So when I get to the, I'll go into a little bit more detail about the knowledge exchange. So there are more than 2,000 resources related to knowledge exchange, which could involve policy, action plans, different resources. And then in terms of the data hub, there are more than 300 data layers. And these will, I'll go into that in a different slide, which I probably also won't be able to see. And then when it comes to connecting stakeholders, there are more than 550 members, and 40 of those are GPML partners. So in terms of the tools available in the digital platform, oh yes, I can see. Right, okay, so the tools available in the digital platform. So when it comes to knowledge exchange, so knowledge exchange is really providing the information available. So there is a library which has, wow, I really still can't see. There is a knowledge library and lots of case studies and learning centers. So I do encourage you, the GPML digital platform is free. It's open to everyone. I also have handouts at the back, which gives you more information, as well as a link for you to go explore. In terms of the connecting of stakeholders, there are different levels of stakeholders. So you can have general members, but then you could also have experts. So the experts are there also to sort of fact check what is being uploaded, because everyone can upload, but because we want this database to be used for things such as action plan development and other things like that, you really want the data to be accurate. So there will be some fact checking of data with the community, and also trying to get people connected. You can share events and also engage via forums. When it comes to the data hub, there are different levels. There are various analytics and statistics. There's also a glossary and also story maps. I really encourage you to, even though you might not be as focused or as interested in marine litter, there are things there that can be of value to you, but it could also be of value for you in terms of data management, because they might not necessarily be related to marine litter, but there could be things related to nutrients, or even currents. So there are some interrelationships that could be relevant, even though you aren't specifically focused on marine litter. And there is also a help center. There are weekly check-in desks, which are sort of working as tutorials to help you use the digital platform. And there are lots of other resources. So we are in the phase 4 release currently. And one of the things that is involved, one of the things that I actually like, is that there's a personalized workspace, not only for action plan development, but also there's a lot of information available on this digital platform, so it can get quite overwhelming. So having this personal workspace, you essentially can go on and say, this is what you're interested in doing. So whether it be action plan development, it could also be that you're an academic and you're focused on something related to microplastics in a specific environment. So there are lots of filters where you can sort of modify what is given to you and get in. It could be for specific regions or specific habitat types. So there are a lot of layers that you can apply, and the personalized workspace really is great for that. And it's also simple, a little bit simpler, and also very user-friendly. And it's really trying to encourage so you have trying to encourage engagement between all the different stakeholders, and also data harmonization, because everyone is collecting data. Every time there's a beach cleanup, people are collecting data. And then sometimes nothing really happens with the data, or sometimes everyone's using different methods. So really trying to get data harmonization. So when combined, you can have amazing data sets that sort of combine different geographic and temporal factors. And also there's capacity development. So there are training, so through the digital platform. So I will say that the Global Partnership on Marine Litter have a massive open online course as well. And so there's a lot of training to develop capacity in different aspects, but also sharing knowledge and sharing lessons learned, and also training to also help develop. In terms of the simplified experience, each individual that joins can have an individual account. So even though there are different types of membership, they can be individual, organizational, and even on the country level. Each individual that joins can create their own account. There's also a new landing page, which makes things a lot easier to understand, but it also focuses on what the plastic pollution issue is and how to promote engagement. There's also a centralized menu to make things a little bit easier to access. And then also as I previously said, this personalized experience is a new aspect that is being piloted within within phase 4. So I went too fast, but thank you for joining me for this. There are several little handouts. So this one is really about the Global Partnership on Marine Litter. And then I also slightly presented a poster, although I was running around. There is also copies of this poster available. So I would encourage you, whether as an individual, whether as a lab, whether you have an influence at your country, if you want some of these resources, you can take it back with you and I encourage you as an individual to join and start exploring the digital platform, and also encourage your organization or even your country, if you have that level of influence, to also join. And thank you. And I'm sorry about my blindness. And I think I do have time for questions.
Thank you so much, Fadila. Very interesting questions for her on the GPML. Oh, you got one, Marta?
No. Oh, we do.
Thank you, Padilla. This is a really nice talk, perhaps a really nice initiative. An initiative that in the region we are needed so much. Maybe not a question, but more like a comment. Because from the MPA perspective, we also are trying to compile data and information and for analysis, for determining parents, and provide guidance and recommendations for do more effective MPA management. But it turned that collecting the data and overcoming perhaps legal frameworks that different countries have, all internal protocols for sharing the data can become a barrier. So it is a recommendation, but perhaps you can consider a joint effort with other networks that are in the region that are trying to collect data. So we approach the focal points at countries on a more coordinated way. Thank you.
Thanks, Marta. And just as a little point of clarification, I'm presenting this on behalf of the global effort. So this what I presented is not something I've worked on individually. This is a global effort to so this is on a more global effort. On the regional effort, we have looked into sort of developing database. And I do agree that there is need to sort of integrate across different aspects, MPAs and other issues. So I hear what you're saying, and I think it is something that we should consider. But on the level of the global partnership on marine litter, it's unlikely that they are going to branch off into marine protected areas. And those are the aspects, because what they're trying to cover right now is already so broad when it comes to the whole aspect of marine litter and pollution prevention. But it is something that I recognize that we do need for the region. I think we have a follow-up.
Yes, I'm not just referring to join on the same platform or whatever. What I'm referring is to analyze legal frameworks for sharing data. That's it.
Okay, thank you, Marta.
All right, excellent. Thank you so much, Fadila. I hope all of you go and check out the GPML if you haven't already. And we will move on to our next talk. We have two more. Can make it. We're almost to lunch. Is Jennifer Stein, who's going to talk about estimating coral population losses across the Florida Reef Tract due to stony coral tissue loss disease.
Thank you. And thanks for everybody letting me come to this wonderful fisheries conference and talk about coral. So shifting gears a little bit, again, thank you. I'm Jennifer Stein. I work for Florida Fish and Wildlife Research Institute in the Florida Keys. And I work in the coral program, obviously. But this presentation is going to basically summarize the impacts of stony coral tissue loss disease on the stony coral population. It'll also provide a little bit of insight towards how dramatic losses in coral population directly affect our ecosystem health and function. Now. So for those of you that aren't familiar, stony coral tissue loss disease is a virulent coral disease that most often results in full mortality of a coral. It was first documented in 2014 off of Miami-Dade County, and it rapidly spread to the northern extent of the Reef Tract, and then gradually south over several years through the Florida Keys, eventually ending up in the Dry Tortugas in May of 2021.
It currently affects over 20 of our species here in Florida. And recent assessments of the disease up in Southeast Florida, in the Broward County area, indicate that we lost 80 to 90% of the population in certain species. So it's pretty devastating. So again, having these large losses of these major reef building colonies, you can assume that we're really going to see large impacts to not only the reef structure, but a lot of that complexity as well. So now that the reef tract has, or the entire reef tract has been impacted by this disease, reef managers, reef researchers, even the media are left asking, what did we actually lose? How lethal was this disease? So previous large mortality events have typically been estimated through % coral cover, which is a good spatial estimate of what we've lost, but no one has really looked at how the actual population changed due to this event. So the objective of this study was to model and quantify the total number of corals lost due to this disease. So to answer this question, I utilized data from 2 different types of programs. The first type of program was a fixed long-term monitoring program. So it's a repeated measures. And this type of program is best used to estimate changes in population over time. The other type of data we used was a random site data collection. So this type of data is better used to look at spatial distribution of conditions. So despite these different site allocation methods, they both have a similar method of collecting coral demographics. So combining these different types of data provides this unique opportunity for us to look at the change in a population at a specific scale, and then extrapolate that to the greater reef tract to look at the whole system impacts. So I totally forgot to switch to this slide. That was the slide I just talked about. So prior to our analysis, we decided to focus on 7 SCTLD susceptible species. Those species are here up on the screen. I'm gonna spare you all the names because they're long. But besides the one that's directly in the middle, all of these specific species are known to be major reef builders. So then to define our study area, we divided up the reef tract into 4 different subregions, as seen here on the map. Then we further stratified the reef into cross-shelf habitats. We did exclude the dry tortugas from this analysis because while we're putting this all together, it's still kind of in this epidemic phase. So we won't be able to assess the loss until a couple of years after that. So to model and quantify the total number of corals lost to this disease, we performed 3 different steps. We first took the data from the fixed site monitoring programs, and we calculated a pre-disease and post-disease density. Then we could calculate a rate of change between those 2 densities. Then taking that rate of change, we go to step number 2, and we calculate pre-disease densities with the random site data, multiply that by the rate of change to get a post-disease density. Then in step number 3, we take the pre and post-disease densities that we calculated in step 2, and we multiply them by the total area of each subregion habitat. Once we have those total abundances from the pre-disease and post-disease, we could subtract them to get a total loss of coral colonies. So again, the reasoning behind using these 2 different types of programs is that the fixed site monitoring program is better for estimating temporal trends, while the random site data are better for estimating spatial patterns. So to perform step 1 of the study, we first needed to determine when these pre and post phases took place. So because the disease took several years to move throughout the Keys, well, this is important, so I'm glad I switched the slides. Because the disease took several years to move throughout each one of this subregions, we had to determine when those took place in each one of the subregions. So looking at the slide here, each one of these figures is a different subregion. So Southeast Florida in the upper left to the upper Keys, middle Keys, and the lower Keys in the bottom right. So along the x-axis, we have years. And on the primary y-axis, we have total abundance of colonies, and that's the blue bars. And on the secondary y-axis, we have prevalence of disease. So the yellow bars are all diseases combined, and then the white bars is just stony coral tissue loss disease. In each one of those figures, you see a dashed line. That's at 5% disease prevalence. Anytime a disease goes above that 5 disease threshold, we consider it as an epidemic phase. So looking at all these figures, you can see a clear progression of the disease through these different subregions. And looking at the brackets, you can see we can determine that way which is the pre and which is the post disease. And we excluded the epidemic years from the analysis. So using these pre and post densities, we can then calculate a rate of change. So after we calculate those densities and get our mean rate of change, we can plot those, and that's what I have here on this figure. So this figure shows the change rate for each one of the 7 species. So along the x-axis are the 4 subregions, and along the y-axis, we have the mean rate of change, where anything below that dashed line means there was a decline in the population. Each one of the dots is a habitat within each one of those subregions. So I wanted to pick out one just to kind of go over you that this is a proportional scale. So here we have Dicasenia in Southeast Florida on the inner reef, and it's at about a 0.15 on the proportional scale. That equates to an 85% reduction in density for that species in that habitat. So looking at the 2 corals in the red box, those are in the Mandrinidae family. They're the most highly susceptible. And you can see that the majority, can I go back? The majority of those are below the 0.1 level, and that equates to a 90% reduction in density. So one thing to also take note on here is a lot of geospatial differences between not only the species, but within species, those geospatial differences between the habitats and the subregions. So once we have that rate of change that we calculated in step 1, we take that and we multiply it by the pre-disease densities we calculated in step 2 from the random site data. Once we multiply those, we can get the pre and the post densities. This is an illustration of the upper Keys, where each one of the rows is a habitat and each one of the columns is a different species. Again, I highlighted the Mandrinidae family, where you can see again, between the pre and the post, we have about an 80 to 90% reduction in density. So for step 3, in order for us to extrapolate these density values that we calculated, we need to first know the total area per subregion habitat. So we calculated this in ArcGIS. And once we had a full acreage, or square meters, we were able to determine how many 10-meter squared transects we could fit within those habitat groupings. So as an example here for the Upper Keys, it was about the Upper Keys patch reefs were about 46 million square meters. That equates to about 4.6 million 10-meter squared transects. So knowing how many 10-meter squared transects fit into these subregion habitat groupings helped us better be able to extrapolate the data from the densities to get our total abundances. So once we extrapolate those densities to get the pre and post abundances, we can then subtract the post from the pre to get a total loss. So in this figure, it's set up the same as the last one. We have the subregions along the bottom, but along this y-axis is estimated total loss in millions of colonies. And again, each one of the columns is a species. So first off, again, I've highlighted the Mandrinidae family because they are the most susceptible, and your eyes are really drawn to the Dicasenia where in the Upper Keys Patch Reef, we saw it was a 4.5 million corals lost. And just to point out a few more, Copophilian natans was once really abundant on our patch reefs in the Keys, and we saw a 35 million colonies lost. Oh no, that was 2.5 million colonies lost. So this is a really eye-opening figure. I mean, to think about, these are in millions of colonies, not thousands, not hundreds. So this is a really substantial amount of our population lost from this disease. So in summary, this study provides reef managers, reef researchers, other stakeholders with the first assessment of quantitative loss of what we lost from these species. And these losses are going to be significant because, like I explained when I outlined the species, these species disproportionately provide coral cover and structure. These are our major reef building colonies that we are losing millions of. So since this study really only targeted 7 of the 20 species known to be susceptible, there is inherently many more corals lost than what we've outlined in this study. So it's really hard to comprehend losing millions of colonies, but these numbers are really needed for managers and restoration practitioners to really set restoration targets and also help them understand the infrastructure that's going to be needed to perform these restoration efforts. Nope. So the impacts of these dramatic coral losses was not only substantial for the coral population, but the impacts to the benthic community and the fish populations is largely unknown. We don't know how this is going to influence them in the future. But of the programs that I talked about that we use for this analysis, the fixed site monitoring programs actually do also collect benthic cover data. So we can use the benthic cover data collected by these programs to basically understand the role of coral abundance with community or with the change in benthic community. Similarly, NCREMP, the National Coral Reef Monitoring Program, that was one of the random site programs, also collects data on fish community structure. So that data can be used to identify connectivity between the reef habitat and those coral losses and how that affects the fish population. So these findings are only possible because these important, very valuable long-term monitoring programs are in place. And therefore, I would like to thank also my co-authors for this, Rob Ruzicka, Mike Colella, Stephanie Schottmeyer, and Colin Shea, our statistician, and all the programs that were developed, FWRI's DRM program and the CREMP program, Nova Southeastern's CCREMP program, and then NOAA's NCREMP program. Without these programs, none of these analyses would be able to take place. And with that, I'll take questions.
Thank you so much. Great job, Jennifer. I guess we could take one quick question. Somebody else? Oh, hold it.
The losses are so staggering here. And it's honestly, quite frightening. It's so important to get this quantified. What I do wonder is, working with stakeholders, have you had issues of when you just get these huge numbers, like 3.5 million coral colonies lost, does it become incomprehensible for folks to digest? And do you have ideas for how to kind of communicate, what this might actually mean in terms of losing a whole foundation species for an ecosystem?
As a biologist, it's often tough for me to communicate a lot of things as an outreach type of material, things that put things in context where I can communicate them to the public. And yeah, this is going to be a really hard, bit of data to convey to the public to help them understand what's actually happening. I mean, most people that you talk to in the public or that I have talked to don't even know this disease exists, and may not even be able to tell the difference between what a dead coral is and a live coral is. It's just the reality. So I haven't thought of a way to convey this information. I'm mostly the scientist that collects it, but it's a very important task and definitely one that's going to take a lot of thought to tackle. And I know a lot of the restoration practitioners have a big hand in this and outreach and trying to promote the restoration of our reefs, but it's definitely going to have to be a dynamic approach to it.
Do you take images and videos when you're doing your data collection?
Yeah, so the long-term monitoring programs, we take regular videos. We also do structure from motion. So we have a lot of different types of documentation, but the long-term repeated measures, we do a lot more of that imagery work, whereas the disturbance response monitoring is meant to be a rapid assessment of coral condition. So we're mostly just focusing on the coral, doing a quick survey to look at their health, and not really doing a lot of the benefit community and things like that.
Thank you.
All right, thank you so much, Jennifer, for that somewhat depressing talk. All right, our last presenter for the morning is Crystal Hightower, who's going to talk to us about the rapid expansion and subsequent decline of lionfish in the Alabama artificial reefs.
Hi, thank you so much for having me. I'm Crystal Hightower. I'm the research coordinator with the University of South Alabama's School of Marine and Environmental Sciences. I'd like to acknowledge my co-authors, our PI, Dr. Sean Powers, Monica Powers, Craig Newton, and Dr. Bob Shipp. So red lionfish, as many of you are familiar with, are an ornamental Indo-Pacific reef fish, extremely popular in saltwater aquaria. They made their first appearance here in Florida in the 1980s on the Atlantic coast and moved rapidly south and westward to the warm waters of the Gulf of Mexico by the 2000s. So they threaten Gulf of Mexico reef environments by direct predation and then through indirect interactions by competition. So they have year-round reproduction and then compete for resources with native fishes. So while red lionfish in the northern Gulf of Mexico are still data limited, we do have some of the best researchers here in our backyard examining life history, movement, and potential controls on their invasion. I'm definitely not an expert on red lionfish at all, but we all know Alex Fogg here from Okaloosa County, who has spent much of his career investigating these fish. So looking at distribution and length frequency, reproductive life history, age and growth, and then also Kristen Dahl and Will Patterson looked at movement, home range, and depredation via acoustic telemetry. And then also Harris suggested epizootic population control in the northern Gulf of Mexico via this ulcerative skin disease that we do see. So an area I do spend most of my time studying is the Alabama Artificial Reef Zone. So Alabama Marine Resources Division, as a part of the Alabama Department of Conservation and Natural Resources, has invested so much time and resources in adding and maintaining the Alabama Artificial Reef Zone. So it began back in the 1950s by the Alabama Charter Fishermen's Association in offshore Alabama and has expanded into what we see here on the left. So for a small fee and upon approval of the design, you can deploy your own private fishing reef. And these zones now exist in a nearshore zone, just offshore in 3 to 6 mile zones, and then also further out around 12 to 70 miles offshore. So some of these examples are on the right. They're private reefs. They're mostly chicken transport devices or chicken coops. You can see that on the top right. But then also there are other structures put out by the state, such as prefabricated concrete designs that we see here in Florida as well. These circulatoral reefs, large and small reef pyramids, army tanks, toppled rigs, and many other structures. So Alabama's artificial reef zone is still currently the largest AARZ in the US. So of course, lionfish benefit from this increased habitat availability and productivity in the AARZ. They proliferate in the warm waters with no native predators. And many groups are working to remove these fish from Alabama's reef zones, but removals via traps and hook and line, as we know, are not super successful. So their removals are limited to diveable depths. So we do have a few tournaments in Alabama, not as big as the ones here, to remove these fishes. And then also the Alabama Seafood Marketing Commission has tried to push the popularity of this as a dish. See if I can get this to start. So our lab, the Fisheries Ecology Lab at USA, is part of a long-term fishery independence survey. It's mostly focused on red snapper, but data are collected on all species. So I'll just give you this roundup of our fishery independence survey. So we first side scan sonar to look at contacts. So we've mapped around 33% of the seafloor off Alabama. We have covered almost half of the Alabama artificial reef zone. So then we trawl to look at the forage fish base. So next we use remotely operated vehicles. We have an Outland 2000 ROV, and this is what our main gear type is in seeing these red lionfish. So this is one of those chicken transport devices or chicken coops, and you can see lionfish on it. We don't have many Goliath grouper off of Alabama, but we have observed them. So this just shows the utility of ROVs in this fishery independent survey for examining other species besides red snapper. So our ROV survey is coupled with vertical longline sampling. So we use a bandit style, commercial style reels. We first test water quality, so look at temperature, salinity, dissolved oxygen at depth. And then we use 8, 11, and 15-knot circle hooks. This is a standardized CMAP protocol, and we fish for 5 minutes. So we also have GoPros on the line, so we can look at catch on the lines, things such as gear saturation, and then also depredation events. So we get link frequency data on every fish that we catch, as well as we weigh every fish and get the size distributions that way. Then we take these samples back to the lab to be processed. So we're taking tissue samples, otoliths for aging and growth, as well as sometimes looking at gut contents and then reproductive characteristics. And then last but not least, we also bottom long line to look at the apex predator assemblage and also get some of these large reef fishes. So these are 15-knot circle hooks, and we're following the standard CMAP procedure for bottom long line as well. So we put out a mile-long bottom long line and soak it for an hour. and we tag and release any shark that can be boarded. And then for bony fish like red snapper, we'll take otoliths for age composition and then also other samples just like with the vertical line. Okay, so again, for this purpose for red lionfish, I'm just going to focus on trends in the ROV abundance because we really just don't collect the fish via our other gears. So here it's just a video to show you some examples of just the sheer number of red lionfish at their peak abundance in 2015. So there are almost so many lionfish that we've saturated our video gear here. You can't really even see the reef pyramid in the background. And then this is just an example of another chicken transport device. Okay, so let's get into some trends. On the left, we have lionfish total abundance from the ROV with year on the x-axis and then total count on the y. So although red lionfish were first observed in the Gulf of Mexico between 2006 and 2010, we did not see them in our survey until around 2012. So you'll see the peak in abundance at approximately 6 to 8 per structure in 2015 to 2017, and then a rapid decline since. So this is for all structures inside and outside Alabama's reef zone, just off our coast. On the right, we have average total count by habitat. So now habitat's on the x-axis and then total counts on the y. So it's no surprise that we do see higher abundance of red lionfish on artificial structures. So to get at the number of red lionfish in Alabama's artificial reef zone, we use our side scan sonar data to estimate the total number of natural and artificial structures. So our benthic habitat assessment program includes identifying contacts within these 2 kilometer square grids you see on the left. The purple, green, and blue are inside Alabama's artificial reef zones. And then the areas to the west, that light blue, yellow, and orange are outside our reef zone, but still offshore Alabama. So we then determine the number of contacts, their size, and their designation based on our side scan. And we've covered almost half of Alabama's artificial reef zone. So to model total abundance, we use a simple multiplicative model based on side scan. We estimate around 74,174 reefs in Alabama's artificial reef zone. So this is our total estimate of red lionfish in Alabama's artificial reef zone. So based on the estimate of reefs annually, you can see the peak in 2015 to 2017 on artificial structure, and that's the lighter color. And then this is just like we saw in the average total count, but then we estimate a larger abundance on natural reefs just based on the sheer number of qualifying rocks and natural structure off of our area inside the reef zone. So we estimated a maximum of over 370,000 lionfish in Alabama's artificial reef zone. And then we've just declined to around 67,000 just within this 1,060 square mile area. So this is an example of what we see today. This was last week, actually. You can observe few lionfish on this collapsing coop. We're also working with the state of Alabama, MRD, on an estimate of habitat loss due to degradation of a lot of these reefs. So a lot of the private structures are metal structures, and we do lose habitat over time. So that's another really interesting thing that we need to look at. And one of the questions, like, how is this going to affect the decline of red lionfish in our area? And if that decline will continue. So we'll continue monitoring red lionfish and other species via our red snapper assessments, as well as during the great amberjack count in the coming years. While studying these invasive species, again, is not the goal of this research program, it is an added benefit. And hopefully in this case, we'll continue to see these invasive fish decline. And with that, I would like to thank our institutions, the University of South Alabama, the Dauphin Island Sea Lab, as well as our funding sources from the state of Alabama, Alabama Department of Conservation of Natural Resources, and the Marine Resources Division.
Excellent. Great job, Crystal. Very interesting. Any questions for Crystal or lunch? Not from the audience. I have a quick one.
Yeah.
Do you have any idea of what has caused the decline in the lionfish?
So, they're obviously the ulcerative skin disease could be an issue. We do collect some samples via opportunistic, dive sampling. We've seen some, but not as much as these guys see here in Florida. It could just be the natural progression of an invasive species. So we see this peak in abundance and then start to see a decline. So we don't know, and there's nothing that's really sticking out, but it is coincident with some increases in some of the smaller forage fish face, which is really cool.
Interesting. Thank you. All right. Are there any announcements? Don't forget to get your t-shirts if you haven't already. They're $10 cash. We're meeting back at 2 o'clock. Is that still the case? Yes, at 2 o'clock. So you guys enjoy your lunch. Thank you to all of you for sticking around, and thank you so much to the speakers. And hold on, Joanna has something to say too.
This is the session called Invertebrates and Aquaculture. My name is Megan Davis, and I'll be moderating this afternoon's session. Our first speaker is not here, so that's given us a few more minutes to get started. I'd also like to make just a quick announcement about the proposed ruling that NOAA has put out to list the queen conch as a threatened species on the Endangered Species Act list. And so the comment period had been this week, but they have extended it to December 15th. And they're also having a public hearing, which will be on November 21st. So that's not next week, but the week after. And if you need to find out some more information on that, you should be able to look it up on the internet, looking for queen conch, proposed ruling, NOAA. But if you have any questions about that, I'd be happy to help you along with others from NOAA that are here as well. Okay, so I'd like to introduce our first speaker of this afternoon for the session that's Invertebrates and Aquaculture. Our first speaker is Jewel Thompson-Beneby, and she'll be speaking this afternoon about community-based approach to queen conch management in the Bahamas. Thank you, Jewel.
All right, good afternoon, everyone. I am Jewel Beneby, as Dr. Davis has mentioned, and I'm very thrilled to be with you guys today, sharing on this important work that we've been doing in the Bahamas, in East Grand Bahama, to be specific. And as I begin, I'd like to just take a look at our focus species, which is queen conch, and our community-based approach to managing the fishery. All right, so why are we focusing on conch? Conch is an important source of income for Bahamian communities. It's a staple in our diet. It's also very important to our cultural identity. Overfishing, of course, particularly of juveniles, undermines the sustainability of the resource, and low population levels have made conch fishing economically unviable for many fishers. And when overfishing of juveniles is paired with natural disasters, This poses a serious risk for the resilience of the fishery. In general, consumers are uninformed about the severity of the resource depletion. And this limits the ability for the fishers to capture the benefits from fishing sustainably. The decline in conch stocks threatens the livelihoods of over 9,000 fishers who are considered vulnerable. And they also have limited alternative employment opportunities. So if stocks are to be restored and community livelihoods maintained, there is a need to provide multiple incentives for fishers and their families for changed behavior towards a more sustainable fishing practice. So many of you in the room may be familiar with this iconic species, queen conch, Alicorgagus. It is a marine snail, slow moving, slow growing, and the average age for sexual maturity is about 3 to 5 to 4 years, and it's determined by the thickness of the flared lip. Okay, so through funding from the Inter-American Development Bank and the Japanese Poverty Alleviation Fund, the Bahamas National Trust, in conjunction with the Nature Conservancy Bahamas program, has developed 4 components with a view of increasing the socioeconomic benefits derived from conch and the conch fishery in the Family Islands. And particularly, we've been looking at Grand Bahama in 2 communities, which is McLeanstown and Sweetings Key. So if you are familiar with the Bahamas, you're right off the coast of Florida and north of Cuba. And in our northernmost chain is Grand Bahama, which is zoomed out in that box. And if you see the little tiny red dot on there, that is McLeanstown. And right next door is Sweetings Key, the community we've been working in. So we've been looking at a community-based approach, and we've been focusing on including community input in the selection of trainings and benefits that they would receive throughout the project. All in all, it's to achieve these 3 main goals, which is improving the livelihoods of conch fishers, establishing conch-based management of conch within a marine protected area, and creating a domestic market for responsibly fished conch through sea-to-sail traceability and local branding. So first, we worked to establish a baseline, and this was done through May and June of 2018. The Bahamas National Trust, along with the Nature Conservancy and a group of volunteers, we surveyed the community, 3 different communities, which is McLeanstown, Sweetings Key, and we also ventured into West End, and that's denoted by the stars on the map. And we got 104 responses. And we found that the survey told us that most people in the surveyed areas were highly dependent on fishing for their livelihoods, and a little over half of them were owning motorized boats. We also noted that at the time of the survey, 95% of them had access to electricity and 90% access to running water. We did also note that 59% of them had felt that there was a decreased quality and stability of their household income, and 54 of them felt that it was not easy to find employment. So we found that there was a strong interest in value-added and alternative livelihoods activities, and farming was considered to be most useful as an alternative. When we asked about what potential value-added products they would be interested in, conch pearl collection, exporting conch shells, and conch farming were high on that list. We also note that there was a 63% interest in joining a cooperative, which was one of our outputs at the time. And then we also note that there was an unclear understanding of the sustainability of the conch fishery when we asked for perspectives. Okay. So the figure on the left shows the boundary of the marine protected area that's highlighted in green. And this is East Grand Bahama Marine Protected Area. And the right shows the densities of conch populations. And that's the number per hectare, both in 2019 and in 2021. And this is noted by a red circle around 11 of those repeat sites. We did have other sites surveyed at that time as well in 2021, but we were really focusing on the repeat sites. The total density is reflected in the size of the size of the circles for each site. And orange denotes the proportion of conch that were subadults or juveniles based on the absence of a flared lip. And the blue indicates adult or legally harvestable conch having a flared lip. And then sites with an asterisk indicate adult conch densities exceeding reproductive thresholds of over 100 per hectare. But it should be noted that most of the conch reported as adults at these sites were likely individuals whose shell thickness was still too thin to be sexually mature. And of the 11 sites in red, we saw that there was a decrease in conch densities. 3 of the sites showed an increase, but 8 of them actually had a decline. The survey also showed that there was a decrease in total abundance, as well as a shift in conch structure of the conch populations, with a percentage of conch adult remaining relatively stable, but dramatic changes in the juvenile and subadult in the given areas. So just to clarify, the survey really is indicating that there are still significant conch populations in the southeastern half of East Grand Bahama Marine Protected Area. However, the number of conch observed in shallow sites has decreased. And factors to consider would be Hurricane Dorian, which I'll talk about in a little bit, fishing pressure, and then of course conch movement. All right, so challenges. We cannot discuss this effort without acknowledging the severe challenges that this community faced. Hurricane Dorian directly impacted the McLeanstown and Sweetings Key communities in 2019, September 1st, and the residents had massive losses, both in their families as well as homes, boats, and overall way of life. Additionally, right after Hurricane Dorian was the impacts of COVID-19 on the economy, as well as we went ahead and paused our implementation for about a little bit over a year. So after, well, just before we restarted the project, we surveyed for community buy-in, and we found that from November to December, we had surveys and there was 29 responses for these surveys. And during that time, the residents were still living in between the communities as well as in Freeport or other communities as well. So the response rate was a bit staggered. And we also found that from the survey, from the buy-in, we noted that the community responded that they felt an overwhelming ability to manage their fishery when asked, and then also they felt that they would be able to manage the rules and regulations, as well as they felt that they would be able to actively enforce those rules and regulations of their fishery. Okay, so after we were able to get significant buy-in back into implementation, we went ahead and hosted a sustainable conch and fisheries workshop where we were able to secure pledges from the community, and they said that they were really interested in getting these trainings and benefits from the project. So during that workshop, we were able to provide them with information around conch life cycles, also around the pre and post information for Hurricane Dorian assessments, so the health of their habitat that they are fishing in, as well as current and updated fishing regulations directly from the Department of Marine Resources, as well as information on forming a cooperative. We also looked at alternatives. So early on in the presentation, I discussed how they felt that farming was one of the alternatives that they were really interested in. So we decided that we would work with the communities to provide multiple incentives for the sustainability and alternative livelihoods. And of course, we also looked at value-added product training. As the community expressed interest in farming, we established a community garden in each of those communities, supplied them with seed starter kits, and also raised beds. And the last two photos on the right just shows growth after 1 month. We also provide incentives such as the standard and training certification and watchkeeping training, or also known as STCW, which is really important marine vessel safety training, first aid training, fire safety, and survival skills. And this was particularly, oh, that's it right there. This was particularly important because the nearest hospital to this community is about 83 kilometers away, which is an hour. So just increasing those benefits for them to be able to act quickly in the need of, in the event of an incident. And also there are clinics in between as well. However, they have limited hours as well as limited capacity. This is also a foundational requirement for any new and current boat captains. We also took a group of 4 residents and 1 local government official to the Fishing Association and Cooperative in Naguabo, Puerto Rico in November of 2021, where they were able to learn about growing conch challenges and triumphs, similarities and differences in the conch industry from their Caribbean counterparts. This trip was made possible from partners at the Conservación Conciencia, as well as Florida Atlantic University, Dr. Megan Davis, enhancement and stock enhancement professor there. And lastly, one of our products that we've been working on was conch shell jewelry. And this was really important because we were looking at value-added products. How do we turn waste products from the conch into opportunities for them to increase their livelihoods from fishing the conch? And right now we don't have the date set yet, but it's meant to happen before the end of the project, where they're able to learn those techniques. And some of the members in the community are already carving, but we want to increase that market for them. So working towards that. And lastly, we also did a conch branch pilot with the community where we had a technical training with 10 of the residents between both communities. And we were able to erect 2 of the 6 planned erected pens between both communities. And it was stocked to a density of about 20 conch per pen. We engage with community personnel for monitoring and managing the sites. I do have to put in a plug here in regards to challenges in the Bahamas currently. And specifically in the community, we did have some poaching. And then we also had research permit limitations that affect how much we wanted to do within the project, as well as project timelines. And with that, I will acknowledge all of our partners, specifically the Inter-American Development Bank and the Japanese Poverty Alleviation Fund that was so gracious in supporting this project, even through the adversity, as well as all of the residents that I've been working with, and all of the partners and collaborators you see on the screen. Any questions?
Thank you very much, Joel. We have time for 2 questions. Yes, sir.
Oh, hello?
Oh, sorry, Joanne, go ahead. And then just 1 second. I apologize.
I'm just gonna put it out there. I missed the beginning. Maybe you said it then. I'm assuming that a lot of this will be aimed at export, especially the jewelry, selling it to tourists. How are you gonna deal with the CITES issue? Because yeah, somebody comes in with any of that stuff into Bermuda and it will be confiscated on arrival. Unless there's something that resembles a CITES certificate, which obviously this would get because it's cultured, but how have you planned for that going forward?
Thank you for that question. So there's 2 opportunities. There's a local market, but we do think that the export market would prove to be very viable for it. And I think it's just a matter of exploring the permits and getting those allocations in supporting that export. And again, it would be a finished product when it's exported, so not just the raw shell itself. So thank you for that.
Good evening, and thank you very much for your presentation. What do you think is the key to getting the community so involved? But I still see your challenges there, which is.
So it's a multi-layered approach that is needed. I think having the direct incentives and asking the right questions, but also listening to the community is really important, and finding out what their needs are at the time of interaction. This community in particular has gone through so much regarding impacts and devastation with Hurricane Dorian. So we've had multiple levels of challenges just because of natural disaster. But continued engagement is really important during the project and then also after.
I'm wondering whether there's any interventions being done at the sort of the other end of the supply chain in terms of the aspect of demand for more sustainably caught conch. Because in general, when you're trying to move toward a more sustainable fishery, you want to have consumers who either are going to choose, you know, the species caught sustainably, or even willing to pay something of a premium. I mean, if you take like the example of lionfish, most jurisdictions have been pretty successful in educating consumers who are willing to pay an environmental premium because they recognize that consuming this fish is beneficial. But the problem has been that that premium is often garnered by restaurants and seafood wholesalers and doesn't get down to the level of fishers. So are you looking at this dynamic, and at the same time that you're working with fishers, are you trying to create higher demand for sustainably caught conch?
Sorry, thank you for that. I think one of the major things is having them work together. Many of the fishers are working independently or just in smaller groups, but having them kind of work in more so of a cooperative, and that's something we've been working on. And we actually have a workshop in 2 weeks where we'll be finalizing the paperwork towards the cooperative. So that's just one step in terms of getting them to organize and then be able to have a stronger voice in terms of getting a higher price for their conch. And that's just one benefit. But it all starts with kind of getting them to work together and recognize that.
Thank you, Jewel. Another round of applause for Jewel Beneby. I'd like to know if Ruben Molina is in the audience. Okay.
So he submitted a presentation, but that he's virtual, so there's no voice to go with it. So I think let's skip him, and if he shows up, we can bring him in later.
Okay, sounds good. So we'll move on to Eric Salin, who's going to speak this afternoon about advances in aquaculture of the triple tail. Thank you, Eric.
Thank you. I'm going to give an overview of the research we have conducted in our laboratory in recent years to develop aquaculture of this species, the triple tail. So this is a project that the initial research started in the early 2000s at the lab with Jim Franks and collaborators, and then that was continued with a smaller project in 2011-12, and we were able to restart more activities more recently with a couple of projects between 2018 and 2021. So we have some more ongoing research that unfortunately we are not able to complete some experiments this summer. So, but I'm going to present the update we had to this point. First introduce a bit this species. This is a fish that people didn't really know about, not that long ago, a couple of decades ago, and it has become more popular, mostly in the US in commercial fisheries. I think it is more, it's a species that people know more about in other parts of the, Central South America. There's fisheries landing the species, but in the US, it is more of a recent species, at least in the recreational fishery. There's a recent synthesis that has been done by the Gulf State Marine Fisheries Commission on that species, and those data from that survey, and they show that the landing are really small in terms of commercial landings. The market is not yet very well developed, but it is a growing market because now, as people know more about this fish and the flesh quality, their demand is growing. And right now, it's mostly supplied by imports from other countries. So I think that's for those reasons, I think it's a species that has generated interest for aquaculture. This is just a few words on the life history. This is a fish that's found in at the surface in pelagic. It's a pelagic fish, like more in the surface water, around floating debris, sargassum and fish crab traps and things like that. And there are some studies on biology that have documented the spawning, reproductive cycle, and that spawning occurs in summer, and then the larval development supposedly in pelagic surface water. So it's a species. There's interest to culture that species for a number of reasons. I think now people are aware of the flesh quality of the triple tail, and there were some initial studies of grow routes, and that showed that it's a fish that grows very fast. It's one of the fastest growing fish candidate for aquaculture, almost as fast as cobia in the amberjack range for growth rates. And it looked like it was easy to maintain in culture, so definitely species people were interested in looking into. And in a recent, there was a recent surveys of marine species candidate for aquaculture in the US, and this species came up. It was a species where there was not much data, so it was a species that was still in development to reach technical feasibility. So what's been done so far, I think there's been problem with initially with spawnings that are now definitely getting resolved. In maturation in tanks, in some studies there were some spawns, in some others no spawn. It's very infrequent kept spontaneous spawns in tanks. So it's a fish where there was some work to understand the reproductive cycle in captivity and the spawning in particular. And because the spawns, when there were some, had low or no fertility. Sex identification was a challenge to sex the fish that were brought from the wild as broodstock, because they didn't have, there was no clear sexual dimorphic characters. And of course, there's no published data on larval culture and grow out. There's been a few work by private companies, but nothing has been made available to the public. So essentially, we try to work on these aspects at the lab. I think first we start with reproduction. That's the first step to develop aquaculture, to control reproduction and obtain spawns, variable spawns that can be used for larval culture and subsequent grow out culture. The observations that were made in our work was that some females reached advanced stages of maturation, vitellogenesis, and they're compatible with hormonal induction of spawning. But males were found usually didn't release sperm. So there was the initial trials done in the early 2000s and in 2010 revealed that female could be induced and ovulated and release eggs, very large spawns, but the spawns usually had very low fertility, and it pointed to a problem with the male's maturation, that the male were not spermiating and fertilizing the spawns. So our objective was to try to develop a hormonal induction protocol that would address this problem and assess the quality of spawns and for reproduction. And I'll talk a little bit about larval culture and grow out also at the end. So we tried a number of hormonal therapies that have been used successfully in other species. The gonadotropin releasing hormone treatment, GnRHA, that's the one that had been used before, that worked, that led the female to spawn and release eggs, but as I just said, with no fertility. So we tried another treatment that was a direct stimulation with gonadotropin hormone. It's a hormone that bypasses the brain, which is a target of the GnRHA treatment. And directly stimulate the gonad, and then potentially we were hoping that this would avoid the problem with the, for the males at least, of the lack of response to the gonadotropin, the GnRHA treatment. And also we tried another combined treatment that combined GnRH hormone with another hormone that potentially blocks prevents the inhibition of this treatment. Which is due to dopamine and usually related to stress response of the fish. So this, in our first experiment, we had 5 treatments that had gonadotropin direct stimulation, control with no induction, GnRH treatment, and GnRH combined with domperidone, as I just discussed. And also another last treatment that also had a free hormone sequentially. So the results were interesting. I think there was no X-rays control, not simulated. This is what we expected. There was the direct stimulation of gonadotropin, HCG, led to no response either. Then when we looked at the other 3 that are on this graph, we limited to 3 so the graphs were easier to read. As you can see, the GnRH only led to the same response that had been reported before, which means that there were some egg releases, that's in blue. The number of egg released was pretty large. There were very large egg releases produced by female, but the fertility was nearly 0. Oops, I'm sorry. Fertility was nearly 0. And that doesn't work. Where should I point? Okay. Okay, I guess I should point that way. The fertility was nearly 0, and the hatch rate and subsequent survival was, of course, nearly 0. What was interesting is the treatment where we combine GnRH hormone with an antidopaminergic, which again, this is a treatment that blocks the dopamine, which is a compound that prevents the GnRH to work and stimulate the brain, and then subsequently the gonad to spawn, to mature and spawn. Then when we use that treatment, then the results are much better. There is the fertility is still there, the fecundity is still there, but then the fertility is much increased, and of course the hatch rate as well. So these results suggest that really the domperidone, the treatment with added to the gonadotropin releasing hormone, really solves the problem that with dopamine inhibition, which is dopamine is often produced in situation of stress, is a factor that prevents at the same time maturation, final maturation and spawning in captivity, and also prevents the response to a single GnRH treatment. And so this treatment is recommended to produce viable spawns for aquaculture. The treatment with gonadotropin did not work. Probably this is very often there's an overstimulation with this hormone, and even with when combined with domperidone, I think it cannot really resolve that. So in a second experiment, we refine that this is our more recent experiment. We refine the protocol by increasing the dose of domperidone to block the inhibition for longer. And I'm not going to show all the graph, but essentially this improved everything. More egg releases. We went to like 3 or even 4 egg releases with the 10 milligram dose of domperidone. Fertility was even higher. We had some food in the 80 or 90%, and hatch and survival was better. So it looks like when we are on a way to try to have a protocol for to produce. I'm sorry, I have difficulties with this, but to produce a viable spawn for aquaculture. Then our next stage, once we had those viable spawns, was to look at larval culture. For this, we are less advanced because we have not had viable spawns for a long time. But we have started testing protocols that we had successfully used for other species similar with pelagic eggs, surface eggs, and in a similar environment as a triple tail. In terms of light and temperature and feeding sequence. And based on what we knew of the triple tails, and we applied those protocols to the larvae, developing larvae that we obtained from spawning. So, the feeding sequence is presented on this figure and is a relatively standard feeding sequence with rotifer praise and background live algae, followed by Artemia, nuclei, and weaning to drive the feed. So, we obtained, we did produce some post larvae successfully with this protocol. And you can see figures of the different developmental stages. So at 24 days post hatch, we have nearly a completely metamorphosed juvenile, and the survival was very high initially, so they successfully initiated feeding on rotifers, which is a very good positive result. That means that this is a species that will be able to grow in hatcheries with the standard feeding sequence of rotifers and Artemia. And unfortunately, there are still a lot of mortality during, and that's more during the later phase of larval rearing, during metamorphosis and feed transition. So we are working on these aspects with working on the enrichment composition and the feeding, some of the aspects of the feeding protocol. For the grow out protocol, we had unfortunately only a small number of juveniles, but we did a second test of grow out in recirculating systems. And confirmed the very fast growth rate. You see in 10 months, we reached like 1.7 kilo on average, which is really good. So that's definitely worth pursuing at commercial density. So as in a conclusion, I think, so there's definitely some progress with spawning. We have a method now to spawn single pair crosses on demand and produce multiple spawn and including large high quality spawn. It would be nice to obtain broodstocks that spawn spontaneously. And so I think there's definitely some work to do with the captive culture environment to resolve that. And then, but we have at least a hormonal induction protocol that seems to function relatively well, and can be still improve a little bit, but I think definitely will produce viable spawns. Larval culture is where we put our efforts now to improve the feeding protocol and increase survival. And then we need to do grow out at commercial density and marketing studies, because it's a new species for the US market. So with that, I would like to acknowledge our funding sources and many people who contributed to the project. And thank you for attention.
Thank you, Eric. We have time for questions. Nancy.
Eric. So with the dopamine or whatever the heck that was, that actually made the males release sperm because you definitely got better fertilization, but I thought I saw in the graph that the hatch was lower with that. So, or maybe it was one combination of it or something.
No, the hatch was not lower. There were two treatments with GnRH and domperidone, and one was based, there was a lot of variability. So it was not decreasing spermiation, that for sure. The two treatments that had some fertilization were the treatment with domperidone on that graph. I can show it again. Well, I guess no better.
Yeah, no,
after spawning, we could confirm that there was sperm release, when we checked them in water, which is something that we never had seen before. So it definitely there's more clear. I think. Well, we could not. It was not enough to really quantify sperm production because it's still very hard. They have a very hard. Body cavity, so squeezing to obtain sperm, like compared to other species where you can really quantify maybe with a syringe, and that species is more difficult. But there was sperm production that was induced. That's
well, great. That's good.
Yeah.
Good luck.
Yeah, thank you.
Thank you. We have time for another question. Okay, another round of applause for Eric. Thank you, Eric. Our next speaker will be Virginia Burns Perez, and she'll be speaking about inventory of the lobster camp fishery at Turnef Atoll Marine Reserve, fishing camps, fishing grounds, and fishing gears.
Just trying that out. Good afternoon. I'm Virginia Burns Perez, and I'm the Adaptive Management Program Director for the Turnef Atoll Sustainability Association. And I will be talking about a lobster camp fishery inventory that we conducted at Turnef. So Turnef Atoll Marine Reserve was declared in 2012. It is 30 miles long and 10 miles wide. It's 20 miles off the coast of Belize, and it's the largest atoll in the Western Hemisphere. It's the most biological diverse marine reserve in Belize. Earlier this year, there was a expansion to our replenishment zones. So we had a little bump from about 11% to 27% in terms of conservation zones. There's the general use zones where commercial fishery is allowed, and there's also some special management zones where conch fishery is allowed. And I should mention that in conservation zones, recreational fishery is allowed, catch and release only. So when Turnef was declared in 2012, 1 year after the Turnef Atoll Sustainability Association was created and designated co-managers of this reserve. We co-manage with the Belize Fisheries Department. Tourism activities out of Turnef include scuba and recreational fishery. Turnef is Area 6 under the Managed Access regime since 2016. And the main commodities are lobster, conch, spiny lobster, queen conch, and finfish. There are approximately 860 fishers, licensed fishers as of April of this year. And of that 860, approximately 250 are customary, meaning that they fish Turnef year-round. The three main groups of fishers are sailboat fishers, skiff fishers, and camp fishers. Our sailboat fishers mostly come from the northern portions of our country. And our skiff fishers are mostly from Belize City, as well as the lobster camp fishers. They are mostly from Belize City. So in designing this inventory, and this is something that we really needed to work on just to understand what the fishing camp looks like, how many gears are out there, where are they located. We really needed to focus on 4 main areas. So we needed to map the fishing camps,
So the fishing camps are where these fishers are based during their time while they're fishing out there. We needed to map the fishing grounds. So the spatial area on the lagoon floor where they have either their lobster traps or lobster shades located. And then we needed to quantify these gears, characterize them. And for the lobster traps, we took it one step further and started to tag and register these traps. So this took a lot of coordination. We first started this in 2019 and it went right into the pandemic. So it involved us, calling fishers on the phone, where we were out in the field interviewing them. We caught them when they were in their boat out at sea. We went to their fishing camps. And we conducted these surveys to get an understanding of how many gears they had, what they were made of, what it cost, where they were sourcing their material. And then we also use different tools to do the mapping. Sorry? Oh, I'm sorry. I've been pointing at the wrong. Yeah, so, we went traditional paper and pencil in most cases. For the fishers that we were located, we gave them an option of either sketching their areas on a blank map of the reserve. As you can see in the red box up there, or maybe you can't see that clear, but there's a sketch there. We also used the spatial monitoring and reporting tool, SMART, which is a tool that we use daily for our enforcement patrols. This is a tool that collects all spatial information on our tracks, on our fuel usage, the fishers that we're encountering, vessel information, some of their catch. So we use this tool, where fishers had time and it allowed, we followed them and they showed us exactly where their fishing ground was or where the fishing camp was. We collected that spatial information, took that information and imported that into ArcGIS, where we were able to create a map of all the fishing ground. So fishing camps, these are some photos of what the camps look like out at Turnef. As I mentioned before, this is an area, it's a base of operations for these fishers. All year round, they leave a security land, what's the word I'm looking for? Somebody to take care of their camp, a groundskeeper all year round. And then the fishers would just back and forth from Belize City every 2 days, every 5 days, just to check their pots or their shades, take that lobster back to Belize City. Multiple crews, fishing crews, can stay at one camp. And you will see that the number of camps has fluctuated. Some camps have become inactive because of COVID, things got hard. And so they weren't able to sustain the area that they were using before. So as you can see here, the red dots represents the fishing camps out at Turnef. And in the last surveys in 2021, we've counted 31 active fishing camps. In 2011, this was done by the University of Belize Environmental Research Institute's program, and they counted 25 active camps. So there, it has been increasing, but over the last couple of years, because of COVID, some camps became inactive. With the lobster fishing grounds, again, using the SMART tool, sketches, GIS, we were able to get this very chaotic map. And as you can see, there are a lot of overlaps in terms of the fishing grounds that fishers are using, which means there is a lot of conflict out at Turnef. We have situations where fishers are accusing other fishers of moving around their lobster traps or shades or coming into their area. These are, nobody can own the seafloor. So this is all done by gentleman's agreement. In some cases, we have fishers who their grandfather has used the exact area that they are using now. And we've documented fishing grounds in use for the past 50 years. So what we have so far is 44 fishing grounds, and these fishing grounds are used by 28 fishers. The 28 fishers represent the people who own the gears. So while they might have workers, and so it means there are more people in the fishery, these 28 people are the men who own the traps, the shades, they own the camp, they own the boats. So they are the bosses out there. But if you calculate the crews, the entire lobster camp fishery, you know, it ranges between 84 and 150 individuals participating in this. A fisher can have multiple fishing grounds because none of this is legislated. So this is something that we're starting to work on. So in one case, we have a fisher who has 5 different areas that he's traditionally been using. So all of this has really been contributing to a baseline so that we're able to understand what areas of the reserve they're using and what types of gears they're using. So in terms of the lobster traps, this was very interesting to learn. You know, I myself tagged along on a fishing vessel for 2 days, just sitting on the boat and observing just how this was done, you know, asking questions about how they made their traps. And we learned that these fishers were sourcing the savanna palmetto from the Belize rural area. And also learned that the people supplying these savanna palmetto had to get a license from the forest department. So they had to ensure that all of that was cleared before they could sell these bundles of savanna palmetto for about $25. And this is the material that the fishers use to make their lobster traps. They also use a Santa Maria hardwood frame. So our team was out there measuring these traps, trying to understand what they were using, and we were also tagging. And as you can see, there are a few photos here of the team out on the boat doing the actual tagging. So there are 2 types of traps being used right now. There's a rectangular one, and then there's a trapezoid looking one. So we did measurements, and one of the most important thing to understand was what the escape gap for these traps look like. So they range from 1 inch to 1.5 inches. The OSPESCA regulations, which is a regional body that we all adhere to, recommends 2 1/8 inch. So recently Belize had passed some legislation that we were going to move to that 2 1/8 inch, but we did have some pushback from the fishers, and so that has been delayed. I do think that a little bit needs to go in here in terms of what that escape gap should look like for Belize, because I can tell you, being on that vessel, every time they pulled up that trap, I saw with my own eyes that many juvenile lobsters were still able to get through that escape gap. So with the traps, we did take it one step further and we attempted to tag. And so far we've tagged 50% of the traps out at the reserve. We use these cable ties with unique serial numbers in the first instance. And then we also tried out these aluminum tags that also had a serial number. The importance of tagging out there is because we're trying to avoid ghost fishing. We have seen traps caught up in mangroves or just out there, and we have no idea who these traps belong to. Through this tagging and registration process, we're able to trace who owns what traps, right? And then also in terms of conflict among fishers, you know, people are not able to just easily steal a trap if it's tagged. So when we did this in 2020, you know, we counted 7,152 lobster traps. And as you can see, we've had a 15% increase in own traps so far. But what's actually deployed currently is 7,399 traps, which is still an increase. So there was a 7.5 increase in traps deployed. So this is something in Belize, all you need to do is to be a licensed fisher. We can both have a fishing license, and I can own 50 traps and you can own 3,000 traps. So this is something that we need to pay some attention to. And again, in the recent proposed legislation, the government of Belize did include registering the number of traps you had, not to start with any restriction, but just to start to collect some baseline information. So TASA is a little bit ahead because we do understand the numbers that are in this marine reserve that we co-manage. Lobster shades were a bit different. They are not as standardized as the lobster traps. So fishers are using just about anything that can provide a shade for a lobster. So old zincs, old car bonnets, anything that can provide, which is, there's another aspect of this that we need to consider, because it is pollution. But so this is not something we were able to count the shades and characterize, try to get an understanding of what they were using, but we were not able to tag these because it would have required scuba, which was going to take up a lot of time, and so we didn't have the time to get into that. So in terms of, and that photo, which is a great photo that I just saw in a New York Times publication, shows what that looks like. So that shade is a frame with just a zinc covered, and that's what the fishers are using. So currently we have 6,065 shades out at Turniff, and there was an increase since 2020, a 10% increase. And again, this is something that we're trying to consider proposing and some other design of shades for these fishers to use out at Turniff. So if you guys have any ideas, please let me know. So some of the key takeaways is really that this inventory helps TASA to better understand the fishery. It's an iterative process. We've decided now that we will be doing it every other year because things are changing so quickly out in the field. And we need this information to better manage and enforce regulations. We need to ensure that the traps are removed at the close of the lobster season. We also need this information to manage the lobster in a sustainable manner, from tracking the increase of gears. This will allow us, we will be in a better position to make management interventions with our co-manager, the Belize Fisheries Department. It really does help us manage conflicts because we've had several issues where fishers come into the office and we can refer back to these maps and help them find a solution to that problem. We also have fishers contacting us now, asking us if we can go out and tag their gear. So that's really important in terms of relationship. Like at first they really pushed back, but now they're seeing the value in this. And I just want to mention that with the hurricane last week, our team is still out there doing an assessment. But if there are any losses in gears, we are able to say, and if they say, well, I had 500 traps, we can come back to this data and say, indeed, Mr. So-and-so had this amount. How can we help him? And then, of course, all this information contributes to the traceability work currently being done in Belize. Thank you. Big thank you to all our donors who have support this work. And even bigger thanks to the wonderful team at TASA, who they're all currently back home cleaning up after the hurricane. And this work would not have been possible without them. So I just want to take time to acknowledge them. There's a lot of heart in that photo. So thank you. If you have any questions, any ideas on how to improve this project, please feel free to contact me.
Thank you, Virginia. We do have time for questions. Oh, we have okay, we have somebody coming up to the mic and then the virtual, okay? Is that all right? Oh, then we'll go ahead and ask them all. Yeah, go ahead.
So Virginia Michelle asks, do the traps degrade when they're recovered and stay on land for months?
They so they're removed at the end of the lobster season, which is the end of February. And these fishers are the reason they're removing them because by law they have to remove them, but it's also a time for them to fix them up, change strips of wood, make them sturdier. So they are removed. There is some degradation over time.
Thank you. Fadila, another virtual question? Okay, Prescott, go ahead.
Not so much a question. I've been to Belize and seen the great work that they do. And so I want to encourage, everyone to support what I've seen there. These people work hard, and so many times these things are underfunded. And I think if we want to have the success in the Caribbean, it cannot be this endless struggle to find adequate resources for the work that they're doing, because they're swimming against a stronger and stronger current. So I want to, encourage you, Virginia and Walden, to keep up the good work you're doing, and the same struggles we face in the Bahamas. So I'm hoping that persons are listening and they would make a more conscious effort to support the work you're doing there. Thank you.
Thank you, Prescott.
Any other questions? Go ahead, Martha.
Thank you, Virginia. Very, very nice presentation, and congratulations of all that hard work that you are doing in Belize. It's amazing. So keep doing that. I am from Colombia, and there they are using lobster traps, but at the industrial level. So thousands and thousands, and for many years. When we trying to approach that kind of fishery, mostly their responses is that those lobster traps didn't long last. So I'm wondering if you have information for how long or how durable wooden trap is being in Belize, being used infrequently through the season.
Right. So thank you so much for that encouragement and those compliments. Yes, so we did ask the fishers that, and most of them said that they could get through 2 lobster seasons before they start to repair. And repair might mean just switching out one strip of wood. But generally that same trap can last up to 5 years. And they swear by the Savannah Palmetto, and I guess it's just the wonderful resources that we have in Belize, but that's been working for them.
Another round of applause for Virginia. Thank you.
Thank you.
Our next presentation is going to be virtual by Hannah Mae Malahel, and it's the evolution of the benthic communities of Pigeon Islands and Guadalupe Island, the Lesser Antilles, from 2012 to 2021, monitored by a photo quadrant technique. Thank you, Hannah Mae.
Hello, student at University des Antilles in Guadeloupe. I am pleased to present you today the evolution of benthic communities of pigeon islets from 2012 to 2021, monitored by photo quadrant technique. Guadeloupe has the largest reef barrier in the Lesser Antilles and a rich fauna. This biodiversity represents an economical engine. In numbers, reef fishing represents 25 million euros per year and tourism 62 million euros per year. However, reefs in Guadeloupe are facing major anthropogenic and natural pressures, such as coral disease, rising sea temperatures, and predation. The substrate are becoming more available for microalgae development, which is already optimized by heterogeneous and pulse-rich nutrients caused by the erosion of coast and the management of sewage. In this context, the National Park of Guadeloupe set up a monitoring of benthic communities around pigeon islets in 2012. Pigeon islets are located at 150 meters from the coast of Manandio Beach. It's a protected area by the National Park. The study aims to establish a long-term temporal monitoring and to identify the aggressive factors disturbing the balance of the environment. Through this presentation, we will first focus on the evolution of benthic communities from 2016 to 2021. Then secondly, I suggest to have a decade overview by using the data collected since 2012. 12 stations around pigeon were chosen and permanently delimited in order to place the photo quadrat at the exact same place at every campaign. 19 communities are photographed within a 80 by 60 centimeter frame twice a year during the wet and dry seasons. The present study contains 132 pictures. All the pictures are adjusted in Photoshop software, then we can proceed to the measurement of each element in CPCE software. All bleached or necrotic surfaces were also monitored, as well as coral recruitment. For the statistical processing, trend searches were performed using Spearman-Ranks correlation coefficient and dominance ratio between groups and species over time using Friedman-Ranks analysis of variance. To begin with, I present to you the overall composition of Benthic communities, including all the quadrats that are over the period from 2016 to 2021. The group of Phaeophysae has the highest coverage rate, followed by Scleractinians and algal turf. The group of sponges is the second group of animal with 9.1% of coverage rate. According to Friedman analysis of variance, the order of dominance of organisms groups did not vary statistically significantly from 2016 to 2021. In contrast, the evolutionary trends of each group of organisms show significant results. Scleractinian group has a statistically significant negative trend over the study period, and the Phaeophysae, cyanobacteria, and sponges group have a statistically significant positive trend. In order to have a visual view of the trend, on this chart, we can actually see the drop of Scleractinians coverage rate in 2020, indicated by the blue curve, and the sudden increase of Phaeophysae indicated in orange, the increase of cyanobacteria in red, and the positive sponge trend in purple. 2020 is an important year, as you can see, because of SCTLD. This mortal Scleractinian disease occurred in Guadeloupe in 2020. The death of the Scleractinians has made the substrate available for competitors, hence the further increase in the rate of Phaeophysae coverage in 2020. They are bioindicators of environmental eutrophication. The proliferation of cyanobacteria reflects pollution by organic matter and environmental degradation in terms of pollution. Pigeon islets are an occupied site near a wastewater treatment plant assessed as non-compliant. The presence of sponges can be interpreted as a bioindicator of pollution by organic matter. Some species also indicated a good ecological state of the environment. To better understand the impact of the disease, we will focus on the coral community. The coral community is composed by 15 species, including 13 Scleractinian, 1 species of hydrocorals, and 1 species of Zootaria. The dominant species is Poetes asteroides with 30.1% coverage, followed by Montastea cavernosa and Orbicea flaviolata. The quadrats have species classified as critically endangered and vulnerable by the IUCN Red List, and also was protected by the ministerial decree of April 25th, 2017. Friedman test revealed a change in the order of dominance during the study, and the year of 2020 and 2021 are responsible for this disparity that correspond of the passage of LCTLD. The trends according to the Spearman test show that of the 15 species of coral, 13 have a non-significant trend. The other 2, Argyricia agaricetes, have a positive trend, and Poetes asteroides has a negative trend. We can conclude that the coral population is therefore stable post-disease. On the other hand, the trends change after passage of FCTLD. Out of the total of 13 species of scleractinians in our study, 8 species show statistically significant negative trend. Here I present to you the different causes of tissue damage on the same colony, for example. Necrosis caused by herbivorous fish were found to be very low during the study. It represents only 1% of tissue damage. All necroses have healed. Another major event that affected the coral community is a bleaching episode in 2019. This event is not visible on the trends because it did not cause any loss. It accounted for 16 of tissue damage. It was caused by a rise in sea temperature above the tolerance threshold of 29 degrees for a long period of 122 days. In comparison, in 2018, when there were no bleaching, the temperature only exceeded the tolerance threshold for 40 days. Here is an illustration of before and after passage of the disease, responsible for 83% of tissue damage and responsible for massive mortality. These necroses are not reversible. Before moving on, I would like to show you the example of evolution of quadra number 5, where we can see all the major events with the coral bleaching episode in 2019 and the recovery 6 months later, and then the SCTLT and back. By combining the first result process since the start of the study, 2012 to 2016, and those by the present work, 2016 to 2021, it is possible to have an overview of the evolution of benthic communities of pigeon islets over a decade. From 2012 to 2016, the most dominant group were algae-tough, scleractinians, and phaeophysae. We are facing a reversal because of the present study. I recall that the group of the phaeophysae is the most dominant, followed by scleractinians and algae-tough. The first period of the study did not experience any event affecting the balance of the environment, hence these results. Regarding the evolutionary trend of the group from 2012 to 2016, there is not a statistically significant evolution, except for the species which have a positive trend and which is continued in the second period, as well as a reduction of cyanobacteria. We can conclude that before the outbreak of FCTLD in 2020, the benthic communities of pigeon were quite stable overall. About coral recruitment, in the first half, there were an average of 4 recruits per season. In the second half, an average of 2 recruits per season. To conclude, this monitoring represents a real witness to the events that impact the seabed of Guadeloupe, namely the severe episode of coral bleaching due to the unusual rise in temperature, as well as the impact of scleractinian disease. At the end of the study, only 9 species of scleractinian remained out of the 13. Indeed, among the 75 coral colonies which died during the study, 42 colonies died because of the disease. The recruitment rate of young corals does not compensate for the losses. With a view to improving the monitoring protocol, the establishment of additional products would strengthen the data of National Park of Guadeloupe, facing off the loss of biodiversity faced by the current quadrants. Possibly have comparative data by setting up the monitoring in other reefs of Guadeloupe. We are coming to the end of this presentation. Thank you for your attention and the opportunity.
Thank you, Hannah Mae. Does anybody have any questions? Is Hannah on the?
Yes, I'm here.
Oh, great. Any questions for Hannah? Any virtual questions? Okay. Thank you again, Hannah. We're appreciate your presentation and you being here today. We are actually running a little ahead of time. So we have, oh, there is a question for you that's virtual. So hold on.
Yes.
How long, so Janelle is asking, how long is a season? And could you explain the recruitment? And she says 4 per season seems awfully low.
Yes, okay. Thank you for this question. So dry season is from December to the month of May, and the wet season is from June to November. So yes, in fact, the recruitment is very low, but we have to recall that the quadrats are only 80 by 60 centimeters large. So it can explain the recruitment rate.
Thank you, Hannah Mae. Another round of applause for Hannah. So we are running a little quicker in the schedule because we had a couple cancellations. So what we're going to do is we're going to move the Elasmobranchs up and then do the break after that. So while they're getting organized on that, if there's any additional questions for Jewel, for Eric, for Virginia, or Hannah Mae, you could come up and ask those now. I think we also had one virtual question for Jewel. And Jewel, if you want to come to the mic.
So Jewel, I'll read some of the questions. Lashanti asked, what would you say was the biggest accomplishment thus far? And also, I'll just give you the other question while you're walking up, which says, this is from Michelle Scherer from Puerto Rico. She says, great job starting off with a community-based survey to establish a truly local baseline. Were the local fishers involved in the surveys as well?
Okay. So maybe I'll take the last one first. So the answer to that is yes, the local fishers were involved in the surveys. The socioeconomic baseline surveys they were involved in, and in terms of advising for the habitat surveys, they provided information and context in terms of where was good location for where they're fishing in the East Grand Bahama Marine Protected Area. And then the first comment was, our question was, what was the biggest accomplishment? That's a hard question. It was a lot of good accomplishments, but I think just having the stickability after such a major disaster in the community and having the continued interest in the long-term impacts and, well, not impacts, but long-term coordination and participation in the project is a success. So, yeah.
Thank you for that. Any other questions for our invertebrates and aquaculture presenters? Yeah, Krista?
Jewel, where did you get those lovely earrings and what are they?
I'm so glad you asked, Krista. So I guess I am sporting a bit of a conch look today. So my earrings are a good example of value-added product. This is actually made from conch shell. If you want to take a look, yes, so very fancy, but yeah, just one of the opportunities around value-added products. And it's, again, in the Bahamas, the conch shell is a waste product. You see them piled up on middens, and oftentimes fishers aren't even bringing them to shore anymore because of the weight and the gas it takes to go out in the boat and stuff like that. So it's a product that is just kind of going to waste. So trying to figure out what industries can be created and sustained in the economy to support the whole product. So thanks, Krista.
Okay, we have a couple more minutes. If you have any other questions for Jewel, for Eric, Virginia, or Hannah Mae. Okay, let's give a round of applause for our invertebrates and aquaculture presenters. Thank you very much. And I am going to turn over the microphone to Jeremy. And just to let you know, the banquet tonight is actually in this building, just a couple doors down from here.
The banquet's at 7:30.
Okay.
So I'm Jeremy Higgs. I'm at the Center for Fisheries Research and Development at the University of Southern Miss. I'll be the moderator for the Elasmobranchs session this afternoon. We do have some slight modification with that, but thank you all for joining us and rolling with the changes. Our first presenter today is Anna Millender, and she'll be speaking about the life history of cow-nose rays.
Okay. Hi, everyone. My name is Anna Millender. I'm a graduate student and research associate for the University of Southern Mississippi's Center for Fisheries Research and Development. And I'm going to be talking to you guys today about sex-specific relationships of age and growth of the Brazilian cow-nose ray. Sweet. All right, so to get started, I wanted to give you a little bit of background on cow-nose rays. So they are a highly migratory coastal ray. It's characterized by this triangular shape, brown-gray coloration, and a bilobe snout. They're commonly found in estuaries and over continental shelves, where they can be found in solitary and small groups, or even in these large aggregations, as seen through an aerial survey off the coast of Florida. Within the northern Gulf of Mexico, there are two species, Rhinoptera benacis and Rhinoptera brasiliensis. And from here on out, I'm going to refer to them as benacis and brasiliensis for time's sake. And neither of these rays currently have a management plan within this area. So historically, within the Northern Gulf, it was always considered that only one species, benacis, was residing here. However, in 2017, through a morphometric and genetic analysis, it was confirmed that the second species, brasiliensis, was also present. And this entire family of rays has a highly conserved body plan, which means the only external difference between the two is the number of tooth series that they possess. And so in the top two figures, the one on the left is benacis. They typically possess 7 to 8 of these rows of dental bars, while brasiliensis typically possess 9 to 13. So prior to the confirmation of the second species within this area, brasiliensis was considered to be endemic to about 1,800 kilometers of Brazil's coastline, where they suffered near extirpation from artisanal fishing pressure. However, that range has been extended into the northern Gulf of Mexico and more recently up the eastern US coast. And now overlap with the majority of benacis's range. They are listed as vulnerable on the IUCN Red List, and they have extremely limited life history data available, and most of that is based on benacis. So there is a critical need to conduct life history assessments on this species. So therefore, our objective was wanted to collect brasiliensis from the northern Gulf of Mexico to really produce these baseline life history data assessment or estimates. And for my presentation today, I'm going to be focusing on the maximum observed age to date, how we examined growth through a multi-model approach, and estimating theoretical longevity. Since 2016, we've been retaining rays through established long-term monitoring projects with the Center for Fisheries Research and Development. And then when I became a grad student in fall of 2019, we established collaborators throughout the Northern Gulf. So the green stars on that map indicate the offices of our collaborators. I don't want you to think we're collecting cow nose in the middle of Texas. And so for each ray that we've collected, we've taken a disc width, which is the measure of length between pectoral fin tips along the ventral side. We've taken a mass and identified the sex based on the absence or presence of claspers. And then for external identification, we took those tooth counts that I mentioned earlier. However, there is overlap between the species, and so we verified the identity with the number of spiral valve lamellae. So that bottom picture is an open spiral valve, and we've counted the different folds within that spiral valve. Moving on to the aging methods, our first step was to find an aging protocol for this species. However, there are no age and growth studies published, so we turned to other rhinopterid species studies. However, there were discrepancies between all of those, so we conducted numerous trials to find the appropriate method to age these counters rays. So we found that the best location for extracting centra was along the mid-central portion of the exposed vertebral column. So that top picture that looks kind of like a wand, that's the entire vertebral column. And then we did numerous processing trials, trying to find a different method or trying to find a method that was most appropriate, and found that sagittal cross sections about 0.2 millimeters thick using a Raytech gem saw that were immediately adhered to slides with cytosil produced reliable, repeatable sections that was both time and cost efficient. So next, we aged each of those sections based off assumptions that were found in previous literature. So we assumed that the birthmark was formed on the centra prior to parturition, and then based off of data from another chapter of my thesis, we assumed that parturition occurred in June. And between that parturition date and that first winter band, we assumed that there was 6 months of growth, and that all subsequent bands were laid down annually. We counted the total number of bands present on each of those sections from the birthmark to the last band, and calculated biological age by taking that total increment count and subtracting 1.5 to account for the birthmark and 6 months of growth, and then added the proportion of the year from the last winter mark to the date of capture. We used a multi-model approach to assess growth and used 4 growth models that were commonly found in both Kauno's literature and also other Elasmobranchs. So the 3-parameter and 2-parameter von Burton-Landfie, the Gompertz, and the logistic. And for the 2-parameter von Burton-Landfie, we did provide that size at birth or width at time 0, which we estimated around 412 millimeters based on a previous chapter of my thesis. To compare these models, we used the AIC. So the model with the lowest AIC was considered the best fit of the 4 models, and then calculated delta AIC to see how the remaining 3 models fit our data. So a good fit was considered with the delta AIC under 2, and anything with a delta AIC over 10 was considered a poor fit and an unlikely candidate, and anything in between those two were an okay fit, but not as good as a delta AIC under 2. From that best fit or model of best fit, we did calculate theoretical longevity using the growth coefficient. So some general results, since 2016, we've captured 28 females and 48 males. Our females ranged in size from 46 to 103 centimeters, while our males ranged in size from 36 to 97 centimeters. Females were aged from 0 to 15 years, while males were aged from 0 to 14 years. And a majority of our samples did come from off the Mississippi coast around those barrier islands, and that is indicated by those black stars. Those are the capture locations. So getting into female growth, just to orient you to this axis, on the y-axis is our disc width and the x-axis is age and biological age in years. And these are the results from our 4 growth models. The 2-parameter Von Burton-Landfie was the best fit to our data, with a maximum theoretical width of around 98 centimeters and a growth coefficient of 0.31. Based on the Delta AIC, all 4 models fit our data pretty well, as evidenced by the stacked growth models within the figure. For males, we have a similar plot as our females. And again, the 2-parameter Von Burton-Landfie was the best fit for our data, with a maximum theoretical width of 93 centimeters and a growth coefficient of 0.27. The 3-parameter Von Burton-Landfie also fit our data really well, but however, the Gompertz and the logistic did not fit as well as the 2 and 3-parameter Von Burton-Landfie. We calculated theoretical longevity with all 4 of the models for both sexes, but based off that best fit model, we're seeing an estimate of 11 for females and 12 for males. So in summary, our maximum age observed to date is 15 years, and our 2-parameter Von Burton-Landfie was the best fit model of best fit for both males and females. And from that model, we were able to calculate a theoretical longevity as 11 for females and 12 for males. And although these are very preliminary results, this is the first time that this has been studied for this species, not only in the northern Gulf of Mexico, but within its entire range. And unfortunately, we were unable to capture some of those smaller size classes. So we weren't able to calculate an age at maturity, but we will be continuing to collect these rays to calculate that parameter, as well as updating all age and growth parameters for this species. Additionally, we're also conducting marginal increment analysis to verify annual band pair formation. And as another portion of my thesis, I'll be conducting an age and growth assessment for Bonassus within the same time period and area, and comparing that to Braziliensis. So if y'all are interested in that, I can send y'all a link to my thesis defense. And I'd like to thank the numerous labs, the people within those labs, for all of their time and effort in collecting these rays and providing valuable insight for this project. And I want to thank you for your time and attention, and I'll take any questions.
Thank you, Anna. That was timed pretty well. We have a few minutes for questions if anybody has any.
Hi, I have a question. I was wondering if you see any evidence of hybrids and how could they affect your results? And also, do all the rays, they need to be killed? How do you see like non-invasive aging methods going forward? Thank you.
Yeah, thank you for your question. So unfortunately, we do have to sacrifice the animal to conduct these assessments. But without that baseline data, we won't be able to actually have a management plan, like create a management plan for these species. But once we have that information, we don't have to sacrifice that animal again. For the hybrid question, we have not seen anything personally, but I know that you would have to do a more detailed genetic analysis to see if hybridization is occurring.
Hi, great talk. I think it's really interesting that these animals are being found in the Gulf of Mexico, and I'm wondering if you know of any indication that it's like a continuous distribution of them from Brazil up into the Gulf, or if it's just like two separate regional populations of the species?
As far as I know, they've done a genetic analysis for the southern and northern portion of the Gulf of Mexico. And everything else is based off of capture records. So I'm not sure like how continuous it is, but I know it's found in other areas besides that initial Brazil location and then our area.
Are there any questions from online? We have time for maybe one more quick one, if anybody has a question for Anna. All right, thank you, Anna. Give her a round of applause again, please. So a little bit more deviation from the schedule. We're going to have Angie Hoover speak next. Angie will be talking today about some diversity of shown in the long line survey in the Chandelier Sound.
Okay. Hey, y'all. My name's Angie Hoover. I'm from the University of Southern Mississippi's Center for Fisheries. And today I'm going to be talking to you guys about the shark diversity across 3 regions of the Chandelier Island chain based on historic longline data. So to orient you to our study area, the Chandelier Island chain is a north-south oriented chain located south of the Mississippi Barrier Islands and east of Louisiana. This island chain provides various services in the form of anthropogenic services, such as serving as a barrier against hurricanes. It can also help mitigate land loss by insulating the coastline. It also provides environmental services through the creation and maintenance of the Chandelier and Breton Sounds, which are relatively shallow and estuarine environments that are known to provide habitat for a lot of fish species throughout various life stages, including sharks. So this estuarine environment is evidenced by the presence of seagrass beds. The Chandelier Island chain is the only place in Louisiana where true seagrass beds are present, meaning that there are several species present. However, the distribution of these seagrass beds throughout the island chain is not homogeneous. So seagrass beds are only present on the western or sound side of the island chain. They are not present on the Gulf side. They are also more substantially present in the more northern islands, such as the Chandelier Island and Curlew Island, and the distribution or presence diminishes quite a bit as you go further south. Furthermore, the structure of the island chain diminishes as you go further south. So as you can see, these more northern islands are these longer, more contiguous structures, whereas as you go further south, they become a lot more smaller and fractured. So for this study, we broke this area into 3 different regions. So the first region that I'm going to refer to is the Northern Sound region. It is characterized by its orientation west of the island chain and north of Curlew Island, which is that island where everything south of that becomes more fractured and there's less seagrass beds. This area or this region is relatively shallow and does experience freshwater input. The next region is the southern region. So this region is located south of Curlew Island and encompasses the water both east and west of the island chain. This region is more closely associated with the Mississippi River runoff. So it does experience higher levels of freshwater runoff, nutrients, and turbidity. The third region is going to be the Northern Gulf region, which is on the east side of the island chain, and it is directly exposed to the Gulf and drops off more quickly in depth. So the objectives for this study were to compare environmental variables, species composition, distribution, sex, and life stage among the 3 regions of the Chandelier Island chain. And so this color scheme is going to remain consistent throughout the rest of this talk, with the Northern Sound being denoted by a green color, the Northern Gulf a gray color, and the South blue. Data from for this study were collected from NOAA's Southeast Area Monitoring and Assessment Program, or CMAP, bottom longline survey from 2015 to 2022. USM CFRD is the state of Mississippi's partner for this survey. So it's a shark and finfish survey that is split up into 3 seasons per year, the spring being April and May, summer being June and July, and fall being August and September. There are usually about 12 to 15 stations per season. The gear type for this is a 1.6 kilometer or 1 mile main line that is fitted with 100 baited 15-aught hooks, and it fishes for an hour. We also collect and measure water quality at the midpoint of the main line. So every fish that comes up on this survey is identified to species, measured, weighed, and released. For Elasmobranchs, we also identify the sex and tag it before release. So getting into some of our results, this is the station makeup for this study period. The first 3 columns here are the seasons that we sample within. As you can see, we have relatively even sampling across the seasons. We did have a bit of a dip in the spring due to COVID, and the spring of 2020, we weren't able to sample. So that's why that number is a little bit lower. But the last 3 columns are the 3 regions that we separated out. And as you can see, there's almost twice as many stations within the Northern Sound than the other 2 stations. So that's a caveat to keep in mind throughout the rest of this talk. So getting into our environmental parameters, we've got temperature, salinity, dissolved oxygen on the top, and we compared them within season. So for temperature, the only time we saw a difference was in spring, with the Northern Sound being a little bit warmer than the Northern Gulf. For salinity, as you can see, the Northern Sound was fresher in each season than the other 2 regions. Dissolved oxygen, we didn't see any differences. And then with depth on the bottom there, the Northern Sound encompassed shallower stations, while the Northern Gulf encompassed deeper stations, which is what we would expect to see. Moving on to our catch summary. So we did encounter the most individuals in the Northern Sound, the least individuals in the Northern Gulf. As far as species composition, it was relatively the same among the 3 regions, with all of our usual suspects like black tips and spinners, fine tooth, great hammerheads, things like that. But there were a few species that were unique to certain regions. For instance, the tiger shark we only saw in the Northern Sound, and the sandbar shark and bonnethead were not seen in the Northern Gulf. And this last year is actually the first year that we found sandbars in the Northern Sound. So all shark CPUE, so catch per unit effort, is calculated as the number of sharks per 100 hook hour. And as you can see, there was a bit of a trend of a lower CPUE in the Northern Sound when compared to the Southern region. We then selected 4 commonly occurring large coastal species, including the blacktip shark, bull shark, spinner shark, and lemon shark. And for these species, we calculated catch per unit effort, did length frequencies, and calculated sex ratio. So for the blacktip shark catch per unit effort, as you can see, this species is distributed throughout all 3 regions. We did encounter the most individuals in the Northern Sound. However, if you look at the catch per unit effort, it was higher in the Southern region. However, these catch per unit efforts were not statistically different from one another. We look at catch per unit effort within season. The only time we saw a difference was in the spring, where the Northern Sound had a lower CPUE than the other 2 regions. Moving on to length frequency, we've got females on the top and males on the bottom. So each individual from each region was binned into a 100 millimeter length bin. And then you've got the that dark dashed line is the length at maturity for each species. So as you can see, for both males and females, we encountered both immature and mature individuals, with most of the smaller individuals coming from the Northern Sound, whereas the larger individuals were seen in all 3 regions. So for the sex ratio for this species, we were able to calculate a sex ratio for both immature and mature individuals. excuse me. So for both immature and mature, we found in each region the ratio to be female dominated. However, none of these ratios were significantly different from the expected 1-to-1, which is what you would expect to see if there was the same number of males versus females. Moving on to bull sharks. Again, we saw this species within all three of the regions. We did see the most individuals in the Northern Sound. However, again, the catch per unit effort did not differ among the three regions. Looking at catch per unit effort within season, we again saw a difference in the spring. However, this is most likely just due to the fact that we didn't encounter any bull sharks in the Northern Gulf. So for length frequency, for females, we did encounter one mature individual. The rest were immature. For males, we only encountered immature individuals. Again, most of the smaller individuals were coming from that Northern Sound region, and the larger individuals were coming from all three. Because we only had immature individuals, we only did the sex ratio for immatures, and we found that the Northern Sound was female dominated, while the other two regions were male dominated. However, again, this ratio was not different from the expected 1-to-1. Spinner sharks. Again, the species distributed throughout all three regions, and the catch per unit effort did not differ among the three regions. We did not see a difference in catch per unit effort when compared with in season. However, as you can see, we didn't collect any spinner sharks in any of the regions in spring. For length frequency, for females, we didn't encounter any mature individuals. For males, we did have a couple. Again, the larger individuals were coming from the southern region. And if you look at the Northern Gulf, some of the smallest individuals that we encountered, and only small individuals, were encountered in the Northern Gulf. For sex ratio, all three regions were found to be female dominated. Again, this ratio did not differ from the expected 1-to-1. Moving on to lemon sharks, as you can see, the catch number here is quite a bit lower than the other species. Again, CPUE did not differ among the three regions. When we compared with in season, we did not see any differences. And then for length frequency, we only collected immature individuals, but we only collected females in the Northern Gulf, and we only collected males or we didn't see any males in the Northern Gulf. So to summarize, we did see similar species composition among the three regions, with a couple of unique individual species for certain regions. In general, we saw lower catch per unit effort and smaller individuals in that Northern Sound region, and greater catch per unit effort and larger individuals in the South, with that Gulf region being somewhere in the middle. And so with that, I would like to thank the funding and our colleagues, and I will take any questions.
Thank you, Angie. Looks like we have a few minutes for questions, if anybody has any for her.
Great. I was curious if you guys have ever seen the small tail shark Acarrhinosporosus sporosus in your survey.
I don't think so.
Okay.
Not to my knowledge, no.
We have not caught it.
I was curious because there are some old records from Biloxi, and it's very, very near from your study area.
Yeah.
Thank you.
Yeah.
Any other questions for Angie? Any questions from online? Well, please give Angie another round of applause, and thank you for presenting today. So we have one talk left for this session, and Brian's making his way to the podium. Won't rush you too much. Today, he'll be talking about the devil in the details. Please start whenever you're ready.
Hi, everyone. Today, I'm going to talk about my project titled The Devil is in the Details, Molecular Identification of Guitarfishes Sold as Pez Diablo. Before I start, I would like to acknowledge my founder, Save Our Seas Foundation. So, modified batos, they are also referred as Pez Diablo, Jenny Hanivers, Diablitos, Garadiabolos, etc. And this depends on the country where they are crafted, but they are primarily made of bitos or primarily made of guitar fishes from the genus Pseudobatus or guitar fishes from the genus Rhinobatus. But also skates and stingrays may be used as well. And those are crafted taxidermized specimens that are used to be sold to fool tourists in short towns, but also to be sold as a curio item, as they resemble mythical creatures such as the bishop fish, dragonfish, basilisk, etc. But this is not something new. In fact, the tradition of modifying batos can be traced all the way back from the 1600s. And if you see it from the book De Piscibus by the Italian naturalist Ulisse Aldrovandi, we can, sorry, my mask. We can see one of the first records of a modified bato. In fact, if you see it in the middle picture, we have a modified skate. So it seems that fishers prefer to use batos because the oronasal region. So having the nares and the mouth close together give like a face-like appearance. For example, in the skates of guitarfish. But for example, another Elasmobranchs that is also a benthic species and is kind of similar from those guys. The oronasal region is located at the top of the snout. So the process of making a Pez Diablo consists in usually making two cuts parallel along the vertebra column. So then the rostral cartilage is removed to give the horn-like appearance. And then also the face, the gills are removed too to have the face-like appearance. They are let dry under the sun, and sometimes a coat of varnish is placed. And Pez Diablo are like snowflakes. They are not two alike. So they can come from many different shapes. And this is depending on the creativity of the artisan, of the fisherman. Sometimes the rostral cartilage is removed, sometimes the gills are removed. Again, but the concept is the same. They are modified batoids. So even some people have taken this way farther. For example, Alfredo Garamendi published a book, Los Garadiabolos, and it has a subtitle that reads from Spanish to English, Aliens Who Live Among Us. And if you see the cover picture, we have modified batoids next to spaceships. And he actually claimed that he was attacked by one of those in La Laguna Negra in Puerto Rico. Some toy companies have been inspired by these modified batoids. For example, here a Philippine toy company. And on the top picture, we have a famous surrealist painter, Salvador Dali, holding what it is a modified guitar fish. So, as I said before, those guitar fishes those modified batos are primarily made of guitar fishes. But guitar fishes are one of the most, they belong to the so-called rhino race. And the rhino race are one of the most imperial fishes in the world. And here in the America continent, we have the genus Pseudobatus. We have 6 species occurring in the Eastern Pacific and 3 species occurring in the Western Atlantic. So now you're gonna have to take my word on this because I can spend the whole hour talking about many issues with taxonomy. But what I can tell you right now that taxonomy is not fully resolved. Many species look alike, and misidentification is a common issue where many species co-occur. So for example, here in this slide, we have 7 different species from the genus Sedubatus, and they all look very, very similar from each other. So in Mexico, we have the most recorded number of that genus. We have 8. We have 6 species occurring in the Eastern Pacific and 2 species occurring in the Western Atlantic. But in Mexico, they are targeted for their meat, but also to be sold as pez diablo. And pez diablo are sold for 2 main purposes: as a curio item, but also as alternative medicine. Some people believe that it can cure cancer, arthritis, among other illnesses. In fact, Mexico has a long time tradition of using using animals for medicinal purposes. So just to give you a better perspective, a pez diablo, a 40 centimeters pez diablo in Mexico and at the fish market can cost you $1, but the same size, turn it into a pez diablo, can cost you between $10 to $50. So the income is way, way higher. And if you go on eBay, the prices just rocket. So now of the 8 species that they are recorded in Mexico, 7 are threatened into a threatened category by the IUCN Red List, and only 1 species is just doing fine. So no one has ever studied this trade. We don't really know which species are being turned into pez diablo. So we don't really know which of the 8 species are being turned into a pez diablo. So as you probably guessed, the objective of my study was to be able to know which species are being commercialized, because if we want to do like conservation management initiatives, we really need to know which species are being traded. And this is very interesting in Mexico, like we have at least 8 species. So Mexico City is an interesting location because it's located in central Mexico and it's 200 miles away from the nearest shore. But it's a highly populated city, and where there is high population, there is also high wildlife trade. So from 2021, 2022, I collect 30 pez diablo from different vendors around the Mexico City. I collect some measurements, and again, those are modified batoids, so it's very hard. So even having the whole specimen can be very challenging to ID. Now imagine when there are some of the morphological characteristics are removed. I collect some tissue samples, recorded female, male, total length, but I was what I can do, because some of the key morphological characteristics sometimes are removed. And for example, here we can see that the rostral cartilage is kept, and sometimes it's left. The gill slits are kept. They remove the gill slits. So based on some of key features, it seems that many of those guitar fishes are coming from the Western Atlantic, but also some species are coming from the Eastern Pacific. And I'm going to tell you how I was able to tell apart based on in certain morphological characteristics. For example, in letter A, we have the speckled guitarfish, Sedubatus glaucostimus. This guitarfish is the easiest to tell apart from the other species that occur in the Eastern Pacific. And if you see, it has blue spots on the disc. While, for example, and even when they are converted into a pez diablo, the blue spots are still present, and even really faded, but still visible. And in the specimens from the Western Atlantic, we the 2 species, we have the freckled guitarfish and the shola guitarfish. They have those enlarged tubercles at the tip of the snout. And when fishers leave the tubercles, we can at least tell from which ocean basin are coming from. So now, as I said before, the taxonomy has not been fully resolved. And I have to start from the scratch, because I cannot really trust some of the sequences that have been uploaded at the NCBI, at the gene bank. So this is taken with a lot of collaboration with people from all over Latin America, Central America, to be able to to create this genetic reference library of the 8 species that can be found in Mexico. And with that genetic reference library, I will be identifying my tissue samples that I collect from Pez Diablo. But for now, I can let you to stay tuned about my findings. And if you are more interested to learn more about the Pez Diablo thing, you can check my profile at Safe Our Seas, where I talk more about those modified guitar fishes. But for now, I would like to acknowledge GCFI for the opportunity to present here, my founders, particular Safe Our Seas Foundation, and my institution, and also all the collaborators that without their help, it would be impossible to keep going with this project, but also the Phillips Labs. With that, I will take any questions.
We have plenty of time for any questions if you have any about Jenny Hanovers. It was a great presentation. Looks we have 2 coming up.
That was really cool. I'm wondering if there's regional differences in the amount of Pez Diablo that's being sold throughout Mexico. Like, is Mexico City kind of the hub of the trade, or do you see it in other regions of Mexico as well?
Oh, yeah, that's a great question. I think so. Mexico City, as I said, is like an interesting place because it's located 200 miles from the closest shore. So it literally could be from any part of any place of the country. As in Mexico City, usually all the trade from seafood usually is distributed first in Mexico City and then moves to other states. But Pez Diablo can be found in also in other states. Like if you go, for example, in Guerrero, it's most likely that those species are going to be from species found in the Eastern Pacific, like in Acapulco. But yeah.
Thank you.
That was a great presentation. And this is just a comment. You need to bring some of those for the auction. Your drawing, your artwork is wonderful.
Thank you so much.
We have time for another question or two if anybody has any. All right, well, thank you. And now let's give Brian another round of applause for his presentation.
Okay, so we have finished for today, but we still have the coffee break, and I please encourage you, the cookies are here today. Please eat them, or else give them a home in your stomachs. So yeah, please enjoy the coffee break, and then join us tonight at 7:30. It'll be in Ballroom 1, which is 2 doors down from here, and there will be food. It is a banquet. There will be food. So please join us tonight, 7:30.